Hello everyone. I guess I'm live, right? Uh, so kindly write down in the chat box if clearly visible, audible, everything is good to go. So we will continue. So let everyone join. We will wait for few seconds, and once everyone join, we will continue. Hello Harsh. So is visibility, audibility is good? You can see my screen. Everything is good to go. Can we continue? Kindly respond. Thank you. Thank you, Praveen. Thank you for responding. Thank you so much. So let everyone join. Students are still joining. Okay, so we have started bacteriology today. Okay, and we have started the first portion in the bacteriology, that is gram-positive cocci, the biggest portion, I must say. Right. So we have completed Staphylococcus and Streptococcus and approach to gram-positive cocci. We have studied three type of Staphylococcus and five important type of Streptococcus. So total 8 bacteria from this section we have completed till now. Right. Now I'm coming on the second category. Okay, can I start the second category? Can we start with the second category? Say yes. Can we start? So let me start with gram-negative cocci. So I'm starting with gram-negative cocci. Okay. So in gram-negative cocci, we are having only two bacteria to deal with, Neisseria and Moraxella. So I will teach you these both bacteria here. I will teach you Neisseria also. I will teach you Moraxella also. The point is that Moraxella means two lines are important that I will tell you in the end. It's a very minor bacteria. We don't have to study at all. Moraxella. Two lines I will teach you, but Neisseria is very important. So basically, we are having two Neisserias in our syllabus, two type of Neisserias. Neisseria meningitidis and Neisseria gonorrhoeae. So here we have to study total three bacteria. Hannah, we have to study total three bacteria. So I am teaching you bacteriology. We have started gram-positive cocci. We have finished it. Staph and strap both are done. Now I am starting gram-negative cocci. In gram-negative cocci, I have to teach two bacteria, Neisseria, Moraxella. Moraxella I will teach you, but not at all important, but Neisseria ultra important. Make a 5-star over it. It's very, very important. We will study two Neisseria here, Neisseria meningitidis and Neisseria gonorrhoeae. These are the two type of Neisseria we have to study. So total in this category, we have to study three bacteria. You can say technically two important, two main hero and one side hero. The Moraxella is not important. So let's start with Neisserias. Let's start with Neisserias. So as I told you that there are two type of Neisseria, Neisseria meningitidis and Neisseria gonorrhoeae. Both of them are gram-negative cocci. What is the color and what's the shape? I'm saying gram-negative. They must be red in color. Okay. And they are cocci, so they must be sphere. Hai like pneumococci. I told you pneumococci. They are also in pairs. They are also in pairs. The Neisseria meningitidis is half moon shaped. This is half moon shaped, and Neisseria gonorrhoeae is kidney shaped. See, but again, this is also in pairs and this is also in pairs. That's why this is also known as Diplococcus. This is also known as Diplococcus. So both are in pairs, both are in Diplococcus, both are red color, but the shape is different. This one is half moon shaped and this one is kidney or bean shaped. Kidney or bean, one and the same thing. One more thing is different in them that you must appreciate. Meningitis is capsulated. It is capsulated. One capsule around each pair, like pneumococcus only. Not one capsule around each bacteria. Mind my words. One capsule around each pair. But here in gonorrhea, we don't have capsule. Instead of capsule, we have pili, fimbriae here. So virulence factor here is the capsule. Based on the capsule, it is of 12 type. Pneumococcus based on capsule was 95 type. This one is 12 type. Here, based on the capsule, here we don't have capsule. The virulence factor is the pili or the fimbriae. So these are the differences between them. One more ultra important difference: both are gram-negative cocci, both are in pairs, both are diplococcus. That is the thing. Differences we have seen. This has the capsule. This don't have the capsule. So capsule present here, capsule absent here. Virulence factor here, capsule here, pili. We have seen that. Disease is entirely different. Which disease they cause? So Neisseria meningitidis, नाम में ही है. It causes main disease meningitis. और भी disease है, but main is meningitis. And gonorrhea causes gonorrhea. Gonorrhea causes gonorrhea. Learn the nickname. घर का प्यार का नाम क्या है दोनों का? Neisseria meningitidis को प्यार से कहते हैं meningococci. Meningococci, है ना? And Neisseria gonorrhoeae को प्यार से कहते हैं gonococci. Gonococci. So that is the nickname of both of them. So this is an overview. I hope you got the overview. Everyone got the overview? Everyone? Say yes if you got it. So till now I told you 4 bacteria which occur in pair. The first I told you Streptococcus pneumoniae. Streptococcus pneumoniae. The second I told you is Enterococcus, which was gamma hemolytic. Enterococcus. The third I taught you Neisseria meningitis, and the fourth I taught you Neisseria gonorrhoeae. But the point is that they all are in pairs. They all are Diplococcus. But this one is flame shaped and I'm drawing violet color. Actually, I should draw violet. It is blue now. Let me use violet only. So this is flame shaped and this is in pair. This is spectacle shape and this is in pair. है ना? This one, these two are gram negative cocci. So this is half moon shaped and this is kidney or bean shaped. See the colors I have used so wisely, so wisely. These are 2 gram positive cocci and these are 2 gram negative cocci. You know the difference between the gram positive, gram negative, violet and red. Both of them are cocci. So these are the paired. All of them are diplococcus. Say yes, I'm just comparing. Hai na? So these two we have already completed. Now I'm teaching you these two. Say yes, people. Among them, capsule kahan pe hai? Chalo. Capsule is present in pneumococcus and capsule is present in meningitis. है ना? Tell me the capsulated organism. I will show you in the mnemonic. ये दोनों दिखाऊंगी मैं. Tell me the mnemonic. Mnemonic क्या है? Yes, very, killer, bacteria, double bacteria. है ना? Have pretty nice capsule. Total nine organisms are capsulated. Now in this mnemonic, can you see in this mnemonic? The P is this pneumococcus and the N is Neisseria, but Neisseria meningitis, not gonorrhea. Got it? So every time, whenever the capsulated organism come, I will show you in the mnemonic. Respond. Got it? Anna? So can we proceed? So same thing is given. Currently, I am teaching you the new heading gram negative cocci and Neisseria. The 2 type of Neisseria are in front of you. See the 2 type of Neisseria. I want you to appreciate both are in pairs. This is half moon shaped and this is kidney or bean shaped. If you see the color nicely, they are little bit reddish color. They are gram negative. So diplococcus, meningitis, gonorrhea, half moon shaped, kidney or bean shaped. Here capsule present, here no capsule present. So here around each pair there is 1 capsule, but here we don't have capsule. Here if you say something, we have pili. We have pili. You got this. So many differences you got it till now. Say capsule present, say capsule absent. Say half moon shaped, say kidney shaped. है ना? Here virulence factor is capsule, here virulence factor is pili. इतना तो समझ में आ गया? Everyone got it, है ना? Now culture media. Coming on the culture media of both of them. I'm talking for both. For meningitis also, gonorrhea also. For both of them, culture media are same. On blood agar, both of them show gamma. What do you mean by gamma? What do you mean by gamma? People say no hemolysis. So I can see the colony, but around the colony, no greenish zone, no transparent zone, only colony. Colony के around कुछ भी नहीं है. देखो, no zone around the colony. है ना? That is known as gamma. This is gamma hemolysis. है ना? On chocolate agar, they grow even better. देखो, chocolate में तो और बड़ी-बड़ी colony है. So they grow on blood agar, gamma hemolysis. They grow better on chocolate agar. They grow better on chocolate agar, which is the enriched media. I taught you what is enriched media, है ना? So blood agar, chocolate agar. What is a selective media? हर bacteria का selective media at the tip of your tongue. Mind my words. है ना? Never take my words lightly. If I'm saying something now, it is having weightage. Got it? है ना? So selective media. Selective media you have to learn for all bacteria. ठीक है? So यहाँ पे you have to learn 3 selective media. है ना? Thayer Martin. Thayer and Martin are 2 scientists. They are best friend. Thayer और Martin दोनों ने जोड़ी बनाई और बोला चलो कोई कल्चर मीडिया खोजते हैं, है ना? They have discovered a culture media. Thayer and Martin together. They are best friends, है ना? So what they did, they took a blood agar only. They took a blood agar and they added 3 things in the blood agar. I'm sorry. Vancomycin, colistin, and nystatin, VCN. They added VCN. So all the bacteria are killed. Only the 2 Neisseria survived. So they said it's a selective media for the Neisseria because everyone else is killed. Only the 2 Neisseria are surviving. Later on, they found that few other bacteria are also coming here and surviving. So it is not very selective. It's selective, but not very selective. So they modified it. So the second is the modified Thayer-Martin. What is the modification? VCN to hai hi hai. The 3 drugs are there. Vancomycin, colistin, nystatin. They added trimethoprim also. So the 4th drug trimethoprim is also added. Vancomycin, colistin, nystatin. They added trimethoprim also. If you want to learn the composition, इस पे question आया नहीं है. The question is on the names itself, है ना? Thayer-Martin, modified Thayer-Martin. Thayer और Martin की हो गई लड़ाई. They got a fight, है ना? Thayer and Martin were best friends, but they got a fight between them. So Martin make a new pair with another scientist, Lewis. So the next is Martin Lewis. So tell me the 3 selective media. Say, ma'am, Thayer-Martin. Say, ma'am, modified Thayer-Martin, and then Martin Lewis. First learn the name, है ना? This is a selective media for which bacteria? Say, ma'am, this is a selective media for both the Neisseria. Neisseria meningitis also, Neisseria gonorrhea also. For both of them, the selective media are 3. है ना? What are the 3? Say the names. Thayer-Martin, modified Thayer-Martin, Martin Lewis. Now, question तो इसी पे है in FMG. But in NEET, PG and other exams, sometimes composition is also asked. If you can learn the composition, it's good. If you can't learn, कोई बात नहीं. Don't take load. है ना? जितना कर पाओ उतना करो. But I am telling you. So here it's VCN. Here it's VC. Here it's VC and T, है ना? And here I don't know, please check it Google. मतलब मुझे नहीं पता. You check some book. I forgot to put it, है ना? इसका भी होगा कुछ ना कुछ. So this is vancomycin, colistin, and nystatin. Vancomycin, colistin, nystatin, and trimethoprim. Here I forgot to put in my notes, and I frankly don't want to bluff. I don't know. है ना? Just a second. So why don't you check in some reference, or tomorrow I will check and let you know, है ना? So Martin Lewis, I forgot to tell. ठीक है, चलो. So can we proceed? Learn the names. इन पे बहुत question है PYQ. Learn the selective media. ठीक है जी, है ना? So let's proceed on the unique properties, the biochemical properties. Both the Neisseria are catalase positive. You know what is catalase positive? The bubbles are coming. Name one more bacteria which is catalase positive. Name one more bacteria which is catalase positive. कौन बताएगा? Yes, Staphylococcus. Like Staphylococcus, they are catalase positive. Not like Streptococcus. They are all, both of them. Neisseria meningitis, Neisseria gonorrhoeae, both are catalase positive. Okay, both of them are oxidase positive. Both of them are oxidase positive, but sugar fermentation differentiate them. So here, see here, I will show you the diagram also. Neisseria meningitis, Neisseria gonorrhoeae. देखो सब screen पे देखो, everyone. So I'm saying both are catalase positive, right like this. ठीक है? I'm saying both are oxidase positive, right like this. है ना? So catalase and oxidase cannot differentiate them. So how to differentiate? How to differentiate them? Catalase and oxidase cannot differentiate the two. Hannah, it is common for the two. So how to differentiate the two? The point is that we can differentiate based on sugar fermentation. Let's take two sugar, glucose and maltose. Again, glucose is fermented by both of them. Glucose. This also ferment glucose. This also ferment glucose. So glucose ke basis pe bhi we can't do. Glucose is fermented by both of them. So both catalase positive, both oxidase positive, both ferment glucose. Okay. So there is one more sugar which is known as maltose. Maltose, that will make a difference. M for M. Meningitis for maltose. Meningitis ferment maltose. है ना? Gonorrhea do not ferment maltose. So it is the maltose that can differentiate the two. It's a PYQ, 100 time repeated PYQ. So the two type of the Neisseria can be differentiated based on maltose, maltose, maltose. You got my point? Tell me the unique sugar for pneumonia. You will say, ma'am, inulin. Bank of India का I. याद आया? Inulin वहां का unique है. यहाँ का unique कौन है? Maltose. यहाँ का unique कौन है? Maltose. You will never forget if you learn M for M. M for M. So you will not forget कभी भी. है ना? So meningitis is fermenting the maltose, not the other. Okay जी. Okay. So the same thing is shown to you. Sugar fermentation, it's written here also. So see, meningitis ferment, देखो, glucose and maltose both. And gonorrhea ferment only glucose. The point is that glucose is fermented by both of them. But maltose is a difference. And M for M. Everyone got it? Don't ever forget. Okay? You can see the image-based question can also come. Can you see the 3 test tubes in front of you? 3 plus 3 total 6 test tubes. See? This is glucose. This is maltose. This is sucrose. This is glucose, this is maltose, this is sucrose. The same 3 and 3 sugar, the two pairs. Here I have put 1 drop. Here I have put 1 drop of meningitis in all of the 3. Here I have put 1 drop of gonorrhea in all of the 3. You tell me what will happen. You will say, ma'am, meningitis. See the change in color. Meningitis is fermenting glucose and maltose both, but not sucrose. Gonorrhea ferment only glucose, not maltose, not sucrose. So sucrose is just to confuse you. Sucrose to neither this is fermenting, not that this is fermenting. So remove sucrose. Sucrose to koi relevant hi nahi hai. None of them is fermenting. But it is the maltose. See the second test tube, middle test tube. Middle test tube. See, the meningitis is fermenting, changing the color, and gonorrhea is not fermenting. Say yes, people. Respond. You can get simple image-based question. Ye image aa gayi. They will say identify the bacteria. Ye image aa gayi. They will say identify the bacteria. You should be able to do that. Say yes. So let's revise till now what we have studied. We have studied the two type of Neisseria, Neisseria meningitis, Neisseria gonorrhoeae. Let's revise together. Come. We have seen both of them are gram-negative coca, so I am using red color. And in my PPT, I use these colors very wisely. So here, this one is in pear and half moon shaped. This one also in pear, but kidney or bean shaped. Here we have capsule, which is a virulence factor. Here we don't have capsule. Here we have pili, which is the virulence factor. Based on the capsule, this is of 12 type. And here we don't have capsule. Till now, we have studied that. After that, we have seen the culture media of both of them. So we have seen 3 culture media. Thayer-Martin for both of them. Modified Thayer-Martin for both of them. And Martin-Lewis for both of them. You know the composition. I'm expecting you know for both of them. That is common. Here also we have it. Here also we have it. The culture media are common. After that, we have seen the unique properties. So you will see, ma'am, both of them are catalase positive. Okay? both of them are oxidase positive. ठीक है, both of them ferment glucose. ठीक है, so this is not important. The main point is that this ferment maltose, this do not ferment maltose. Okay, till now we have studied that. Give me a thumbs up. Till now this is the summary. So this is the table ultimately you have to learn. Baki, you have seen the images. Say yes. Now I'm going to tell you the diseases. है ना, ये क्या क्या कराता है? Tell me the diseases. ये क्या क्या कराता है? Tell me the diseases. है ना, and the chapter will be over. So let's come on the diseases of both of them one by one. The most important point here is the maltose and the capsule, which makes a difference. ठीक है? So let's start the diseases. Diseases is separate. So let's start with meningitis. Of course, it will cause meningitis. And let's come on gonorrhea. Of course, it will cause gonorrhea. Disease is entirely different. One is brain ka disease and one is sexual, sex organs ka disease. Disease is entirely different. Let's talk about meningitis first. So the diseases, the pathogenesis of the meningitis, it causes 4 diseases. Number 1, naam mein hi hai, meningitis. So of course it causes meningitis. Number 1. Of course, obviously it causes meningitis. Bhai naam mein hi hai na. So it causes meningitis. Number 2, it causes rashes. It causes non-blanching rashes. Number 3, after going in the brain and the skin, it goes in the blood. It causes meningitis in the brain. It causes rashes on the skin. After that, it goes in the blood. In the blood, it causes septicemia. Septicemia, severe septicemia. From the blood, it goes to multiple organ and involve all the organs of the body. It is known as Waterhouse-Friderichsen syndrome. Say it out. Waterhouse-Friderichsen syndrome. Very important PYQ. The question comes like Waterhouse-Friderichsen syndrome is caused by which bacteria? Students don't identify. Bhai, kya itna bada naam aa gaya? It's very simple. Waterhouse-Friderichsen syndrome is multi-organ failure. It is caused by Neisseria meningitis. It is the most severe complication. So tell me the 4 diseases caused by Neisseria meningitis one by one. Say, ma'am, नाम में ही है meningitis तो कराएगा ही, obviously. Even a kid can tell that. उसमें कोई important नहीं है ना? It causes rashes also. After that, it goes to the blood, causes septicemia, septicemia. And after that, it involves multiple organ of the body. It is known as Waterhouse-Friderichsen syndrome. Say yes, people learn the 4 things. The 4 diseases caused by Neisseria meningitis. Can we proceed? Can we proceed? So learn the 4 diseases. Most important is Waterhouse-Friderichsen syndrome. जिस पे question आता है, repeated PYQ आता है. Remaining are not very important, but you already know that. I mean. So coming on the pathogenesis of gonorrhea now. Gonorrhea. Gonorrhea causes only one disease. It was causing 4 different diseases, but gonorrhea causes only one disease. Only one. The name is gonorrhea. It's a STD, sexually transmitted disease. It's a STD. Today I will teach you 4 STDs. This is the first one I am teaching you. In bacteriology, there are 4 bacteria which cause 3 or 4 bacteria which cause STDs. So this is the first one I am teaching you. It's a STD, sexually transmitted disease. So in all of them, the name of the disease is gonorrhea. Gonorrhea. I don't know the exact spelling, but yeah, it's gonorrhea. Whatever. Okay. Gonorrhea. But the point is that the presentation is different. What are the symptoms in male? What are the symptoms in female? What are the symptoms in girls? The symptoms are different in female, the symptoms are different in teenage girls. And what are the symptoms in infant? So the disease ka naam ek hi hai. The name of the disease is gonorrhea. It is gonorrhea in male, gonorrhea in female, gonorrhea in girl, and gonorrhea in infant. But the presentation is different. The symptoms are different. So let me tell you the symptom in each category. Males start with urethritis. Learn the first organ. Urethritis. The male come to you, doctor, I'm having urethritis. What do you mean by urethritis? Urethritis means pus in urethra. Urethra pe pus hai. So at the tip of the urethra, there is a pus drop and it's very painful. Sexual intercourse is painful. Urination is painful. That will be the complaint of this male. Urethritis. After that, prostate gland is involved. Epididymis is involved. Seminal vesicle is involved. I mean, multiple organs are involved. Don't learn them. Finally, when all the sexual organs in the male are involved, penis ke around the penis, multiple small sinuses are formed and the pus come out of the body. First, internal pus is formed. Then multiple sinuses are formed around the groin area, around the penis, and pus come from that sinuses. It's a severe complication known as it is known as water cane perineum. Water cane perineum. This is the most severe complication. So learn the first and the last. It starts with urethritis, it ends with water cane perineum. Say yes. It starts with urethritis, it ends in water cane perineum, and multiple organs are involved. Learn starting point, end point. ये male में हुआ. I will show you the images also. ये male में हुआ. Coming on the female. In the female, the first organ involved is cervix. I will tell you reason also why cervix. It causes cervicitis. Please learn. It's very important question. It causes cervicitis. Okay, ji. After that, fallopian tubes are involved, known as salpingitis. After that, complete PID, pelvic inflammatory disease is there. After that, multiple organs are involved. In the end, fibrosis occur in the entire peritoneum. Fibrosis, the most deadly combination, complication. It is known as Fitz-Hughes Curtis syndrome. Maybe spelling nahi aati mujhe. Fitz-Hughes Curtis syndrome, but I can identify in the options. It is Fitz-Hughes Curtis syndrome, है ना? So it's not a literature class. तो spelling मत mind करना मेरी. I'm sorry for that. ठीक है? लेकिन the words are right. It is Fitz-Hughes Curtis syndrome. Okay, ji? Fitz-Hughes Curtis. So learn the first, learn the last. So here we start with urethritis and we end at Waterkane perineum. Here we start at cervicitis and end at Fitz-Hughes Curtis syndrome. Okay, ji? Got it? Coming on the girls, teenage girls. Teenage girls, the first symptom is not cervix. It's not cervix. It's vulvovagina. You may be thinking, ma'am, why? Why in females it's cervix? Why in girls and vagina? Why the organ is different? Both are females, no? She's also female. She's also female. She's a married female and she's a teenage girl. Then why you are saying the different organ? Okay, I will tell you. Wait. Okay, chalo. Let me tell you. Chalo. Okay. So let me draw the genital tract of a female. So this is the uterus I am trying to draw. This is the cervix and this is the vagina. है ना? So let me say the females, adult female I mean. है ना? And let me take a girl. है ना? So point is that in adult female, the pH of vagina is acidic. है ना? It is acidic. And in girl, the pH of vagina is alkaline. It is natural. है ना? It's natural. I mean, it happens physiologically. It's not a disease. है ना? ऐसा ही होता है. And after marriage, the pH of the vagina shifts towards acidic. है ना? But in girls, it is alkaline. Now, when Neisseria gonorrhea enters, in both of them, the root is same sexual intercourse. So when through sexual intercourse, the Neisseria gonorrhea enters, just suppose this is the organism. The name of the organism is Neisseria gonorrhea. So Neisseria gonorrhea cannot survive in acidic medium. No organism can survive in acid. Acid mein kaun jiyega? No one wants to survive in acid. So when it enters, if it is a girl, so it will stay here only because it is alkaline and cause vaginitis. So in girls, it causes vulvovaginitis. But in adult female, when it enters, here it is acidic, so upar, it shift upward and causes cervicitis. है ना? I tried. समझ में आया? देखो, समझ में आया तो अच्छी बात है. ठीक है? नहीं समझ में आया तो डबल अच्छी बात है. Learn the summary. है ना? Learn the summary. Nobody is going to ask the reason. है ना? Learn the summary. Got my point? So what I said, I'm teaching you Neisseria gonorrhea. It causes a disease. The name of the disease is gonorrhea. Gonorrhea, the name of the disease. But the presentation is different. What is the presentation in male? What is the presentation in female? What is the presentation in girl? And last, I'm telling you infant. है ना? Males में first and last. First start with urethritis with multiple organ involvement. Last is Waterkane perineum. In females, it is cervicitis with multiple organ involvement. Last is Fitz-Hughes Curtis syndrome. ठीक है? In girls, it's vulvovaginitis. So it makes a sense here. है ना? And you get many PYQs here. So please understand your question very carefully, whether they are asking the adult female or the girl. See, people respond, jage ho, soye ho. Jage ho. ठीक है. सुनो. Listen my point. In male, female or girl, the root is sexual intercourse. So it is coming from the partner, the sexual partner I mean. The sexual partner is giving the disease. Whether it is a male, the female is giving. The female, the male is giving. The girl is giving. And it can occur in homosexuals also. Anyways, coming on the infant. But infant don't do sexual intercourse, obviously. So in infant mein kaise aayega? What is the root in infant, I want to ask? What is the root? What is the root? Okay, what is the root here in infant? Infant don't do sexual intercourse, obviously. So how does it enter in infant? That is my question. Does anyone know? Shall I tell? Infant mein karayega kya, wo baad mein batati hu. First tell me the root in the infant. Then I will tell you the disease. Listen, let me draw. Yes, very good. Listen, let me draw a pregnant lady. The uterus of a pregnant lady. This is the cervix and this is the vagina. Okay? She is pregnant. So this is the fetus inside the uterus. And this is the placenta connecting the fetus and the mother. Okay? Now this lady is pregnant and she is having gonorrhea. So where does it present in adult female? Adult female mein kahan pe hota hai? It is in cervix. So this is gonorrhea. Can you see? Here is the organism present. Neisseria gonorrhea is present in the cervix. She is having cervicitis. During pregnancy, she is having gonorrhea. Ideally, it should be treated before delivery. The drugs should be given, the antibiotic should be given, and before delivery, it should be treated. But this lady is from some village, some periphery, some village, and she has not taken even a single antenatal clinic. She has not attended, and she is doing home delivery. There are many females in India like this. They don't take even a single visit. They become pregnant and they do the home delivery directly. So she is infected and she is not treated during the pregnancy. Now the time of the delivery arrived, and she is doing vaginal delivery. So when the child is coming out of the vagina of the mother, these are the eyes of the child. So this organism, Neisseria gonorrhea, enters in the eyes of the child and causes a disease known as ophthalmia neonatorum. है ना. तो चाइल्ड में क्या होगा, ऑफ्थेल्मिया न्यूनोटरम. सो व्हाट इज द रूट यू से, द रूट इज इन्फेक्टेड बर्थ कैनाल ऑफ द मदर. बोलो एक बार, व्हाट इज द रूट, से इन्फेक्टेड बर्थ कैनाल ऑफ द मदर. एक बार फिर से बोलो, इन्फेक्टेड बर्थ कैनाल ऑफ द मदर, दिस इज द रूट, है ना. इट इज नॉट थ्रू प्लेसेंटा, माइंडेड, है ना. समटाइम यू गेट द क्वेश्चन वर्टिकल रूट, नो, प्लेसेंटा के थ्रू नहीं आ रहा है. इन्फेक्टेड बर्थ कैनाल. One more thing you have to notice here, it will occur only in vaginal delivery when the child is passing the cervix or the vagina. If you do cesarean section na, the child ऊपर से ऊपर आ जाएगा, newborn. In cesarean section it won't happen. It happen only in vaginal delivery. I hope you all got it. I hope, है ना. So that is the fourth presentation. See the presentation now. Everyone see the presentation now. Everyone here with me. So in males, learn urethritis. Start with urethritis, followed by prostate, seminal vesicle, epididymis, and finally Waterkine perineum. Learn the first, learn the last. In females, the first is cervicitis. It starts with cervicitis. Then fallopian tubes are involved, PID, and finally Fitzhugh-Curtis syndrome. ठीक है. In girls, it's vulvovaginitis. है ना? It's vulvovaginitis, not cervicitis. And in infants, it's ophthalmia neonatum from the infected birth canal of the mother during vaginal delivery. If you wish, you can write not CS. Not CS. है ना? LSCS में नहीं होगा, है ना? Cesarean section में नहीं होगा. I hope you all got it. ठीक है. So that is gonorrhea. This is ophthalmia neonatum. You can see the eyes have the pus and the child can turn blind because of that. Ophthalmia neonatum. We are done. So you want me to revise it again? We are done with the 2 chapters. Neisseria meningitis भी हो गया, Neisseria gonorrhea भी हो गया. We are done with Neisseria. Gram negative cocci are completed. Gram positive also completed, gram negative also completed. We will revise very quickly. चलो, morphology बताओ. Both are diplococcus. Both occurs in pairs. This is half moon shaped. This is kidney shaped. Here we have the capsule as a virulence factor based on which it is 12 type. And here we have pili. We don't have capsule here, we have pili. Okay, it's like this. This is the morphology. After that, we have seen the 3 culture media, which is common for both of them. In both of them, there is Thier-Martin. In both of them, there is modified Thier-Martin. And in both of them, there is Martin-Lewis. So culture media is common for both of them. After that, we have seen both of them are catalase positive. Both of them are oxidase positive. Both of them ferment glucose. But the most important thing which we have to note here is maltose. This one ferment maltose, this one don't ferment maltose. After that, we have seen the diseases. So let's talk about the diseases here. Let's talk about the diseases here. Here the first disease is meningitis. है ना? After that, rashes. है ना? After that, it goes in blood known as septicemia. Okay, ji. And finally, Waterhouse-Friderichsen syndrome. Don't forget. ठीक है. Here only one disease, gonorrhea, not multiple disease, only one. But the presentation is different in male, in female, in girls, and in infants. Okay, ji, है ना? So say in males it's urethritis and finally Waterkine perineum. In females it's cervicitis and finally Fitzhugh-Curtis syndrome. In girls vulvovaginitis and in infants ophthalmia neonatum. The root in these 3 is sex, sexual intercourse I mean. And here the root in infant is infected birth canal of the mother during vaginal delivery. I am done. And now all the important points I have shown you here. Now basically question comes on the complication. You get the question on Waterhouse-Friderichsen syndrome, Waterkine perineum, Fitzhugh-Curtis syndrome. Please, these are the catchy words. You have to pick it up. The catchy words. Now you can get confused between the water. This is Waterhouse-Friderichsen syndrome and this one is Waterkine perineum. Don't get confused. I mean, ठीक है? So all that drug of choice for both of them लिखा है notes में. You can read. It's third generation cephalosporin. Third generation cephalosporin. Both of them, है ना? Especially ceftriaxone. Ceftriaxone you take. बाकी doses you will read in pharma. Ceftriaxone you take. Here we give the drug only to the patient. Here we give the drug to both the sexual partner, whether the partner is symptomatic or non-symptomatic. If the husband is coming, give the treatment to the husband and ask to give to the wife also. I mean, whatever this is sexual partner. And if the female is coming, ask the female to give the treatment to the sexual male also, because otherwise it will remain in the couple. है ना? If you treat the husband, the wife is a reservoir. If you treat the wife, the husband is a reservoir. It will be within the couple. Can we go? Can we go ahead? We are done with the Neisseria. Read the question. Tell me the answer. चलो दोनों Neisseria हो गए. दोनों Neisseria हो गए. Something in questions they are asking about lab diagnosis also. You are not telling. Medicos, whatever they will ask, you will be able to find out. No? So lab diagnosis, just read it. Sample, maybe I should tell you the 2 types of Neisseria. Neisseria meningitis, Neisseria gonorrhea. So Medico is asking, I asked you to read the charts parallelly now. In the charts, there is a column lab diagnosis, which I am not telling you right now. So wherever it is required, I will tell you. Otherwise, you can understand. है ना? उसमें कुछ ज़्यादा ये नहीं है. Meningitis में we will take CSF as a sample. है ना? Because it is causing meningitis. Here, if the male is there, we will take urethral discharge. In the female, we will take cervical swab. In the girls, we will take vulvovaginal swab. In the infants, we will take the discharge around the eye, है ना, eye का discharge लेंगे. So sample will be different. After the sample, you will do the morphology. यहाँ पे भी तुम्हें डिप्लोकोकस मिलेंगे, यहाँ पे भी, बाकी कहानी तो सेम है. ठीक है, you will do the culture और वो सब तो सेम ही है. ठीक है, nothing important. चलो, read the question, tell me the answer. First. Meningococci differ from gonococci in what? What additional thing they have but they don't have? ऐसी चीज बताओ. They are intracellular. They possess a capsule. They ferment glucose. They are oxidase positive. What is the correct answer, people? Yes. So capsule is something unique present here but not here. I forgot to tell you they both are intracellular. You can write down somewhere. Both of them are intracellular. They are preferably present inside the neutrophils. Neutrophils of human body, in the human blood. Both of them. So intracellular are both. Glucose को भी both ferment करते हैं. Oxidase positive भी both है ना? लेकिन capsule is present in meningitis, not gonorrhea. Learn both are intracellular. ठीक है? Okay, you can see this is a neutrophil, this is a neutrophil. है ना? You can see the multi-lobated nucleus, and inside which you can see the dot-like organism. Both are intracellular. Read the next question. Waterhouse-Friderichsen syndrome is seen in. Waterhouse-Friderichsen syndrome. What is the answer? Waterhouse-Friderichsen syndrome, obviously it is meningitis. The most severe complication, है ना? That is Neisseria meningitis. Okay. The next question. Differentiating feature between Neisseria gonorrhoeae and Neisseria meningitis. Differentiating feature between Neisseria gonorrhoeae and Neisseria meningitis. Can you please tell me what is the correct answer? Is it lactose fermentation, maltose fermentation, mannitol fermentation, or sucrose fermentation? Yes, obviously it's maltose. M for M. Neisseria meningitis, Neisseria gonorrhoeae. So this is maltose positive, maltose negative. I mean, this ferment maltose, this do not ferment. So maltose will separate the two. Don't get confused. Mannitol nahi hai. M for maltose hai, not mannitol. Please learn that. Okay. So we are done with gram-negative cocci also. So till now, two sections are done. Gram-positive cocci and gram-negative cocci. Before moving to the third large section, let me tell you something. There is no elevator to success. You have to take the staircase. I mean, don't look for the shortcuts. I've seen most of the students, they always look for the shortcut. कि मैम टेल मी अ वे, टेल मी अ बुक, जैसे तैसे बस 150 आ जाए उसकी तैयारी करा दो. डोंट लुक फॉर द शॉर्टकट. अल्टीमेटली यहाँ शॉर्टकट मार भी लोगे तो लाइफ में कोई शॉर्टकट नहीं है. यू वांट टू बी सक्सेसफुल, यू हैव टू टेक द रूटीन रोड. देयर इज नो शॉर्टकट फॉर सक्सेस. ऑलवेज रिमेंबर, इट्स अ रूल, इट्स अ यूनिवर्सल लॉ. जैसे न्यूटन लॉ है ना, ग्रेविटेशनल लॉ है. यू कैन नॉट चेंज अ लॉ, यू कैन नॉट चेंज अ लॉ. इट्स अ लॉ. इफ यू वांट टू बी सक्सेसफुल, यू हैव टू टेक द लॉन्ग रूट. देयर इज नो शॉर्टकट. ठीक है ना? सोना बनना है तो तपना तो पड़ेगा. हीरा बनना है तो कटना तो पड़ेगा. यू अंडरस्टैंड हिंदी? तो एक बार तो तपना और एक बार तो कटना पड़ेगा, तब तो वो निखार आएगा. ठीक है? सो यू आर वर्किंग लाइक करंटली. यू कैन अंडरस्टैंड व्हाट आई मीन टू से. एनीवेज, चलो लेट्स मूव अहेड. ठीक है, चलो लेट्स कम ऑन द नेक्स्ट टॉपिक. वी आर डन विद gram positive cocci. We are done with gram negative cocci, and now I'm starting the third category, that is gram positive bacilli. So let's start the third category, gram positive bacilli. We have to study six bacteria here. How many bacteria? 6. One by one we will study. जिसमें से कि 4 तो थोड़े बड़े हैं, 2 small आते हैं, जिसमें कुछ खास नहीं है. But 4 are थोड़ा सा बड़ा, average, not very बड़ा. ठीक है? Moraxilla not important. Doxycycline. Moraxilla you will study in ophthalm maybe. Moraxilla पे doxycycline is asking. Moraxilla पे only one question comes. You can write anywhere. It causes a disease known as angular conjunctivitis. ये ऑफ्थेल में भी पढ़ोगे, है ना? लेट मी ड्रॉ आई. आई फॉरगॉट टू पुट द इमेज. है ना? आई का जो एंगल्स होता है ना, एंगल ऑफ द आई पे कंजंक्टिवाइटिस कराता है. यहाँ पे. सो रेडनेस इज एट द एंगल्स. एंगुलर कंजंक्टिवाइटिस. जस्ट लर्न. इस पे एक ही क्वेश्चन आता है. ओके जी, कैन वी प्रोसीड? सो लेट्स स्टार्ट ग्राम पॉजिटिव बेसिलाई, द 6 बैक्टीरिया. So I will teach you all six, A, B, C, D, L, M. You know what is A, B, C, D, L, M. The sequence may be different, but I will teach you all 6. Let's start with Bacillus. Can I start with Bacillus? Let's start with the first. I'm starting with Bacillus. ठीक है? First I will teach you Bacillus, then I will teach you diphtheria, then I will teach you Clostridia, and then the remaining 3. ठीक है? चलो let's start with Bacillus. शुरू करेंगे Bacillus फटाफट, without wasting any time. Let's start with Bacillus. ठीक है? Bacillus anthrax. Here in Bacillus, I will teach you 2 Bacillus. One main hero, one side hero, as usual. So the main hero here is the Bacillus anthrax. And the side hero here is the Bacillus cereus. है ना? Cereus. So Bacillus anthrax causes a disease known as anthrax. It's very deadly disease. Very deadly. Treatment भी नहीं है ज़्यादा. But Bacillus is a simple food poisoning. It causes simple food poisoning. So I'm going to teach you both, है ना? This I will teach you in detail because it's the main hero here, and this is the side hero. We will see few lines of that. Main heroes के सारे chart हैं. If you wish chart में पढ़ो, if you wish notes में पढ़ो. It's your choice. Let's start Bacillus anthrax. Can I start? You people with me? You can see me. You can see my expression. You can see my gesture. Imagine in front of me only one screen, and I can see your chat box. That's it. It's too boring and monotonous teaching for hours in an empty room. You know, nobody is there, and I'm speaking constantly for 10 hours. Hana, so chat is the only way I can interact with you. So be interactive all the time. Okay, chalo. So let's start with the Bacillus anthrax. Okay, so let's start Bacillus anthrax. You tell me morphology. Morphology of Bacillus anthrax. Whenever I ask for morpho, you have to comment on three things. Capsule present or absent, batao. Huh? I'm asking for Bacillus anthrax. Tell me the morpho of Bacillus anthrax. Capsule batao present or absent. Spore batao present or absent. Motility batao present or absent. So capsulated organism ka mnemonic we know. Yes, very killer bacteria have pretty nice capsule. We have two B, no? So one of the B is Bacillus. So yes, capsule present. I mean to say capsule present. Spore mein the mnemonic is BSc chemistry. BSc chemistry. So again, B is Bacillus anthrax. So yes, spore be present. So you will say, ma'am, capsule also present, spore also present. But if you see the motility में 6 plus 6 दो थे ना, तो if you remember the mnemonic cute baby sleep, है ना, तो cute baby sleep का भी B Bacillus है. But I told you all Bacillus except the main hero, Bacillus anthrax. कल check करके आओ. All Bacillus are motile except anthrax. I taught you this yesterday, है ना? So motility absent है. दूसरे Bacillus में है लेकिन इसमें नहीं है. My point is that capsule present, spore present, motility absent. ये याद करने की जरूरत नहीं है. Visible in the mnemonic, है ना? Don't mug up. If you know the mnemonic on spot, you can calculate. If I ask Bacillus anthrax में capsule है कि नहीं बेटा बताओ, तो you will say, ma'am, just wait. I will apply in the mnemonic. Yes, it's coming in the mnemonic. It's capsulated. I will ask you, tell me whether spore is there or not. You just apply in the mnemonic. Yes, it's spore forming. In the motility, it's non-motile. So Bacillus anthrax के बारे में we have learned three things. What are the three things? Bacillus anthrax, you will say, ma'am, capsule present, spore present, है ना, and motility absent. No need to learn. Apply the mnemonic. I have written the mnemonic in front of you to prove. Don't learn कि भाई present absent. अच्छा, कैप्सूल की एक खास बात है, yesterday भी मैंने बताई थी. All capsule, दुनिया के हर कैप्सूल 9 बैक्टीरिया में है ना, 9 के 9, they are made up of polysaccharide except Bacillus anthrax where it is made up of polypeptide. Yes, where yesterday also I taught you this. It is an exception. I know, so it is an exception. Everywhere capsule is made up of polysaccharide, but here it is made up of polypeptide. I told you the name of the amino acid also. Which amino acid? It's polyglutamate. Polypeptide है तो कौन सा amino acid? Polyglutamic acid, polyglutamate, whatever you say. ठीक है, so this is the unique thing about the capsule. ठीक है, spores. Spores में I taught you yesterday two type of spores. कल थे आप लोग मेरे lecture में और have you watched the recording? Yesterday I taught you two. Just a second. Yesterday I taught you two types of spores, bulging, non-bulging. याद आया? So this is having spore. लेकिन comment भाई bulging है कि non-bulging है? You always have to say. So you say, ma'am, it's non-bulging. And it's like this. It's like this. So this is the bacteria and the spore is inside that. It is not bulging. It's flat. Say, ma'am, it's non-bulging. It's non-bulging and it's central. It's not subterminal, terminal, it's central. So ये खास बात है spore की, है ना? So learn the unique feature of the capsule, learn the unique feature of the spore and non-motile. Motility तो कोई खास बात है ही नहीं. So learn the three things. Capsule की खास बात क्या है? It is polypeptide. Very important PYQ because all other capsules are polysaccharide. This is polypeptide polyglutamate. ठीक है. दूसरा spore की खास बात क्या है? Is it bulging or non-bulging? Say, ma'am, it's non-bulging. It's central. See the diagram, है ना? And it's non-motile. So the same thing is written in front of you. You can say, ma'am, it's non-motile. We know that. Capsule is there. Highlight the word called polyglutamate. Polyglutamate, if you don't understand, you can write polypeptide. And spores are there, write down. It's non-bulging and central. Non-bulging and central. And one more thing I would like to tell. See here in the diagram. Everyone see the diagram, what I'm explaining. I want to explain you a concept. They are one behind the other like the bogies of a train. One behind the other like the bogies of a train. They are one behind the other like the bogies of a train. I repeat, it is known as bamboo stick appearance. Bamboo ka stick dikha hai? So they are one behind the other. One behind the other. This typical appearance occurs only in this bacteria, Bacillus anthrax. Bacillus anthrax is bamboo stick. One behind the other like this. They make a train, one behind the other. Hai yes. Boxcar appearance also you can say. Okay, please learn this appearance. Okay. Now, I'm going to teach you 5 culture media. On all 5, believe or don't believe, there is PYQ. On all 5. Make 5-star name, 10-star over this. You usually get questions from this topic. The culture media. एक एक करके I will show you the images one by one. First, we will start with nutrient agar. We will start with nutrient agar. On nutrient agar, I told you, na, it's bamboo stick. It's like the bamboos, multiple bamboo interlock with each other. The multiple bamboos. Can you see? These all are multiple bamboos. The multiple bamboos are there. The multiple bamboos interlock with each other. इसे कहते हैं interlocking. It is looking the curly hair of a girl. The name of this girl is Medusa. Medusa is a girl having a curly hairs. My hairs are not curly. So Medusa is a girl having a curly hair. So it is known as Medusa head appearance. What it is known as? Medusa head. Learn on nutrient agar, it's Medusa head appearance. ठीक है? ठीक है? It is known as on nutrient agar. Please learn the word Medusa head. Medusa head. Always imagine a girl with curly hair. Medusa head because of interlocking of the bamboos. Say yes. Okay. On gelatin swab, let's take gelatin. Let's take gelatin. You know what is gelatin? Let's take gelatin in a test tube. See, this is gelatin. Gelatin is a semi-solid, like agar. Put 1 drop of Bacillus anthrax here. What does the Bacillus anthrax will do? What does the Bacillus anthrax? Bacillus anthrax start liquefying the gelatin. Like, what is the shape? It's inverted triangle like this. Can you see? It is known as inverted fir appearance. Fir. This is fir tree. Fir tree yani Christmas tree. Christmas tree, the fir tree. But it is ulta, inverted. Can you see? Inverted triangle. So the liquefaction is inverted like this. You must ask him why this is the shape, typical triangular, inverted triangular. Inverted fir appearance, so you got it. But why? Liquefaction occurs in this shape, typically in this shape. You tell me, yesterday I taught you, you will give the answer. Yesterday I taught you obligate aerobe. बताए थे क्या obligate aerobe कितने होते हैं? What was the mnemonic? कल का पूरा आज रिवाइज होगा. Yesterday I taught you 10-12 mnemonics, वो बार बार आएंगे अब आज. Yesterday was your foundation day, ना? हाँ, तो क्या थे? There were 4 bacteria which were obligate aerobe. Mnemonic क्या था? Nesty, pest, must, breathe. याद आया? So breathe is Bacillus anthrax. It's a strict aerobe, obligate aerobe. So you tell me, maximum oxygen is where? You will see my maximum oxygen is at the top. That's why maximum liquefaction at the top. As you move deeper, the oxygen reduce, and that's why the liquefaction reduce. So this is the reason why maximum liquefaction at the top. So inverted fir appearance का reason है obligate aerobe. It's also a p-vacuum. Everyone got it? So please understand. So on nutrient agar, say Medusa head. On gelatin swab, say inverted fir appearance. On a solid media containing penicillin. On a solid media containing penicillin, it's string of pearl appearance. आप एक solid media ले लो, जिसमें penicillin डाल दो. So with penicillin, all other bacteria will kill, only this will survive. Bacillus anthrax. And it will form the colony like the moti, pearls. String of pearls, मोतियों का हार. String of pearl appearance. Beautiful, white, pearly, whitish, shiny colony. String of pearl appearance. ठीक है? Next ले लेते हैं fourth one. The fourth one. Let's take a stain. उस स्टेन का नाम है मिथिलीन ब्लू. हैव यू हर्ड अबाउट मिथिलीन ब्लू? एक स्टेन होता है, एक डाई होता है. मिथिलीन ब्लू इज अ डाई, है ना? सो यू टेक मिथिलीन ब्लू and mix the methylene blue with your bacteria. What is your bacteria? Bacillus anthrax. When you mix the two, methylene blue with Bacillus anthrax, when you mix the two, please understand, when you mix the two, what does methylene blue do? Methylene blue separate the bacteria and the capsule. Separate the bacteria and the capsule of the bacteria. दोनों को separate कर देगा. See the diagram. Make a smear. See the diagram. Where is the bacteria? You see my bamboos. I can see the bamboos like this. Appreciate the bamboos. ये दिख रहे हैं? Bamboos दिख रहे हैं मुझे. See the image very clearly. ये सारे bamboos हैं. अब बोलोगे मैम ओके bamboo तो दिख गया. Where is the capsule? Capsule gets separated from that. And where is it? It is forming clumps. The bluish amorphous clumps. है ना? So bacteria gets separated and capsule gets separated. It is known as Mac-Faridian reaction. It is known as Mac-Faridian reaction. Say yes. So Mac-Faridian reaction occurs when we mix methylene blue with Bacillus anthrax. When we mix the two in the Bacillus anthrax, the bacteria and capsule get separated. The capsule form the amorphous clump, और इसे कहते हैं Mac-Faridian reaction. How many of you got it? Respond. Got it? Very important question. Mac-Faridian reaction. ठीक है? The last one is the selective media for all bacteria, 25 bacteria. Selective media at the tip of your tongue. इसके बाद मैं सबके रिवाइज कराती हूँ अभी तक के जितने पढ़े हैं. ठीक है? The selective media is PLET. PLET. है ना? If you want to learn full form, it's okay. If you don't want to learn, it's also okay. है ना? P stand for polymyxin. L stand for lysozyme. E stand for EDTA and T stand for thalassacid. So it's PLET. PLET media. Okay, जी, है ना? We have studied many selective media till now. Can we revise from the beginning? Who was awakened from the beginning? Step Aureus का बताओ. स्ट्रेप्टोकोकस पायोजेंस का बताओ, ठीक है। उसके बाद कौन सा पढ़ा? उसके बाद कौन सा बैक्टीरिया पढ़ा? निजेरिया, दोनों, निजेरिया मोरक्जिला एंड निजेरिया घोनोरी, साथ में ही है ना, दोनों पढ़े। एंड नाउ वी आर स्टडिंग बेसिलस एंथ्रेक्स, टिल नाउ वी हैव स्टडीड दिस, है ना। सो स्टार्ट सेइंग, हाँ यहाँ पे क्या था? लुडलाम था एक तो, दूसरा कौन था? मैनिटोल सॉल्ट, याद आया? मैनिटोल सॉल्ट। और तीसरा क्या था? कोलंबिया, ठीक है। यहाँ पे क्या था? क्रिस्टल वॉलेट एंड पीएनएफ, क्रिस्टल वॉलेट ब्लड अगार एंड पीएनएफ ब्लड अगार। यहाँ पे क्या था? थियर मार्टिन, मॉडिफाइड थियर मार्टिन, मार्टिन लुइस, है ना। मैंने सबकी कंपोजिशन तक बताई है। एंड बेसिलस एंथ्रेक्स में आई एम टेलिंग यू पीएलईटी. Now to learn the composition or not, it's your choice. I have told you, better to learn. If you can afford learn, don't take a chance. ठीक है? But if you can't learn, it's okay. At least learn the name. Name तो याद करो. ठीक है? So this is what we have learned. So continue the list till end. ठीक है जी? So that is the culture media. So the selective media is PLET. So what we have learned? Can we revise what we have learned? Please tell me the media of Bacillus anthrax. Culture media of Bacillus anthrax. चलो start saying. Start saying. Nutrient agar पे क्या मिलेगा? What do we get on nutrient agar, people? What do we get? I'm repeating, Shivam. Hello, I'm repeating. Wait. So what do we get on nutrient agar? What do we get on nutrient agar? Jaldi, jaldi, fast. Say, ma'am, Medusa head appearance. Medusa head appearance. Let me draw a girl, a beautiful, cute girl for you with a curly hair. Hana, curly hair. So I want to say the curly hair, Medusa head appearance. Okay, ji. Gelatin swab. On gelatin swab, what do we get? Say inverted fur appearance, because it is a strict arrow, inverted fur appearance. On a solid media containing penicillin, the string of the pearls, मोतियों का हार, है ना, methylene blue. So Shivam, when we mix methylene blue with Bacillus anthrax, है ना, let's take Bacillus anthrax in a test tube. So this is the bacteria Bacillus anthrax. I know that, है ना. I'm mixing methylene blue with that. Put few drops of methylene blue. Once you mix the tuna, what does this methylene blue do? बैक्टीरिया में से बैक्टीरिया और कैप्सूल को अलग अलग कर देगा. Now you try to make a smear. What do you get? You will get the bacteria separately. These are the bamboos. ये पूरी bamboos है. And where is the capsule? Capsule अलग हो गया. And capsule is forming the clumps. The capsule is separate. Bacteria is separate. This is known as McFaradian reaction. Got it? So this is McFaradian. Here you get McFaradian reaction. And selective media is PLET. You learn the full form. I am done. So these all are your PYQs. Okay, ji. Can we proceed? समझ में आया? Shivam, others, everyone. Okay. Yes. So let's proceed ahead. This is the culture media. Now let me tell you the pathogenesis and virulence factor here. Chalo. Let's start. सुनो मेरी बात सुनो. Then I will show you in the notes. Bacillus anthrax जो है ना, it's a zoonotic disease. What is zoonotic? What do you mean by zoonotic? It's a disease of animal. It's not a disease of man. Yes, it's not a disease of man. It's a disease of animal. So you will say, ma'am, how does it come to man? How does it come to human? I mean, how does it come to man? Anthrax occurs in human. But my point is that Bacillus anthrax cannot come directly to man. No, it cannot come. It will come to the man via animals only. It's a zoonotic disease. So how does it come to man? Via animal. So let me take a man. You know man, what does man do? Man eats animal. Imagine a man who is eating an animal, who is cutting and eating an animal, who is having anthrax. The animal is having anthrax. So this man will have intestinal anthrax. This man will have intestinal anthrax. In this way, the anthrax enter in the body and it causes diarrhea. That is intestinal anthrax. And by eating the contaminated animal. And the second thing, man wear animal. Wear matlab we wear leather jacket. Do you have leather jacket? We wear woolens in winters. Now winters will come after Diwali. We wear the woolens, scarves, everything. So all the woolen scarf, leather jacket, leather purse, they are made from the animal skin. So imagine an animal who is infected with the anthrax and you are using the product, any product of that animal. So the person can have cutaneous anthrax. This is known as cutaneous anthrax. Okay. This is known as cutaneous anthrax. Anna. Here we have intestinal anthrax, which occurs by eating. This is cutaneous anthrax, which occurs by wearing. And third, man can inhale animal. Inhale. What do you mean by inhale? Imagine who make the sweater. There is a person who is known as wool suitor. Who is a wool suitor? Wool suitor कौन होता है? Wool is obtained from which animal? Sheep. From the sheep we get the wool, and there's a person who suit the wool and makes sweaters for all of us and sell it in the market. No. So imagine a person who's a wool suitor. He is suiting the wool from the sheep, and the sheep is contaminated. The sheep is having the anthrax. So the wool contains the spores, and the person is inhaling the spores. So this person will have pulmonary anthrax. So there are three types of anthrax. Please say intestinal, cutaneous, pulmonary. The root is different. Intestinal occur because of eating animal. Cutaneous occur because of wearing the animal product. And pulmonary occurs because of inhaling the spores. People say something, respond, got it? So these are the three ways. How does anthrax can enter human body? My point is that Bacillus anthrax cannot directly enter in human. No, it comes via animal only. So there are three types. Okay? Now imagine this is a cell. This is a cell. You will say, ma'am, which cell? This is human cell. Human ka cell, human ka nucleus. Which human cell? We have millions of cells in our body. Which cell? You will say the three organs. Either it is intestinal cell, or it is pulmonary cell, or it is skin cell, cutaneous. Anthrax involve only three organs, either intestine or lung or skin. Anthrax do not involve fourth organ. Three organ, either by eating animal or inhaling animal or wearing animal. I learned like this. है ना? So this is any of the three cell. ये human का cell है. Imagine this is any of the human cell. Intestinal, lung या skin. है ना? So imagine this is the anthrax coming. This is the anthrax coming. Anthrax do not enter the cell. Anthrax do not enter the human cell. Anthrax will make a toxin. Anthrax will make a toxin which is known as anthrax toxin. What it is known as? Anthrax toxin, Bacillus anthrax toxin. The toxin will enter. Mind my words. This is bacteria. Let me draw the bacteria with blue color. Bacteria is not entering. Bacteria is forming the toxin. Mind it. The toxin is entering inside the cell. Say yes. है ना? The toxin is entering inside the cell, any of the three cells. After entering inside the cell, it is stimulating an enzyme. The name of the enzyme, in human cell, there is an enzyme. The name of that enzyme is adenyl cyclase. Adenyl cyclase. This toxin stimulate that enzyme. Stimulate, do not inhibit. It stimulate the enzyme. Okay? It stimulate that enzyme. On stimulation of this enzyme, what does this enzyme do? This enzyme basically convert ATP to cyclic AMP. So more and more ATP get converted to more and more cyclic AMP. So basically inside the cell, cyclic AMP concentration increase so much. Because of which the cell will take water. It will become swell, and ultimately burst. So intestinal cell will burst, patient have diarrhea. Pulmonary cell will burst, patient will have pneumonia. And skin cell will burst, patient have pustules. So disease ka presentation may be different, but ultimately in all three cells, cyclic AMP is increased. So hallmark is that, the hallmark thing is that you have to learn. So Bacillus anthrax increases cyclic AMP by stimulating an enzyme, adenyl cyclase, because of which it causes disease. Say yes, people, it's difficult portion, but I'm trying very hard. You may be looking at my efforts, no? I'm trying very hard to explain you. Got it? I'm coming on the diseases, Medico. Wait, here I'm explaining you the mechanism of action of the toxin. So what is the mechanism of action of the toxin? Say stimulate adenyl cyclase and increase cyclic AMP. Listen, how many bacteria I'm going to teach you today? How many? I'm teaching you 25, almost 25, 30 कुछ bacteria है ना, छोटे-छोटे मिला लो तो 30 हो जाएंगे. So 25, 30 bacteria I will teach you. Among them, 10 bacteria secrete toxin. 10 of them have toxin, and toxin will cause a disease. Bacteria will not cause a disease. Toxin will cause the disease. One of them is Bacillus anthrax, जो मैंने अभी बताया. So I want कि मैं remaining भी सारे आपको यही बता दूं. So बार-बार नहीं बताना पड़ेगा. I want कि remaining all these bacteria shall I tell you here only. So again and again, whenever the toxin will come, I have not to teach you. Shall I do so? Out of the 10, मैं 8 यहाँ पे बता दूंगी. Let me see मैंने किया है क्या? Yes. I will tell you 8 here. Remaining 2 बाद में बताऊंगी जब आएंगे. ठीक है? Listen. I'm drawing two cells in front of you. This is a cell. One question pakka from this. Take my words. It's very important. This is a cell. This is a human cell. I have drawn 2 human cells in front of you. है ना? So let me draw the bacteria. This is a bacteria. This is a bacteria. Bacteria is not going in the cell. Bacteria is secreting a toxin. So this bacteria secrete a toxin. This is the toxin. Toxin will go inside. This bacteria also secrete a toxin. And the toxin is going inside. है ना? So here also toxin going here. एक तो मैंने बता दिया. This toxin after going, what it is doing? अभी क्या हो रहा था? This toxin stimulate adenyl cyclase. And adenyl cyclase convert ATP to cyclic AMP, so increase cyclic AMP. And because of increased cyclic AMP, the cell will burst. The cell will burst, the cell will die. So this is the mechanism of action. है ना? And the other mechanism of action, here the toxin, after going inside the cell, it will go to the ribosome. Actually, it will go to elongation factor 2. Elongation factor 2, it will inhibit elongation factor 2. This toxin is inhibiting elongation factor 2. Elongation factor 2 is required for ribosome. No elongation factor 2, no ribosome. No ribosome, no protein synthesis. Since there is no protein synthesis, cell will die. So both the cell will die, but the reason is different. Here the reason Again, I repeat, here the reason is increased cyclic AMP, here the reason is inhibition of protein synthesis. Got my point? So these are the two mechanisms. There are 4 toxins which work with this mechanism. There are 4 toxins which work with this mechanism. Total 8 toxin, 4 plus 4. Got it? So let me tell you the 2 mnemonics. Here the mnemonic is be very efficient boy. This is my mnemonic. Be very efficient boy. Okay? So let me tell you the full form also. There are 2 B, है ना? One is Bacillus anthrax जो मैं पढ़ा रही हूँ अभी, है ना? One is Bordetella, है ना? V is for Vibrio, ठीक है? And E is for E. coli. It is for E. coli. The mechanism of action in all 4 is same. अब मैं 4 बार नहीं पढ़ाऊंगी. Here I have told you ना, जब ये आएंगे तो I will not tell you. You will understand by yourself. I will say a word, it increase cyclic AMP and you will understand by yourself कि yes क्या हो रहा होगा. Say yes, है ना? So please learn the full form. ठीक है? And here what is the mnemonic? Delhi Public Secondary School, है ना? Secondary school or private secondary school, whatever. Delhi Private Secondary School. ठीक है? So what is the full form? D for diphtheria, है ना? P for pseudomonas. ठीक है? S, there are 2 S and S. One is Shiga toxin. One is Shiga-like toxin. Shiga-like toxin यानी virotoxin. ठीक है? Say yes, people say yes. So total 8 toxins हैं. These 4, my summary is that. Listen my summary carefully. These 4 toxins, they all act by increasing cyclic AMP and destroy the cell. And these 4 toxins, they all act by inhibiting the protein synthesis and destroy the cell. So mechanism of these 4 is same, but the cell will differ. Understand my point, है ना? I'm teaching you Bacillus anthrax ना, तो यहाँ पे मैंने 3 सेल बताए, intestinal, lung और थर्ड क्या था, skin, है ना? Vibrio पढ़ाऊंगी तो सिर्फ intestine, E. coli पढ़ाऊंगी तो सिर्फ intestine, Bordetella पढ़ाऊंगी तो सिर्फ lung. Organ will keep on changing, but this is the mechanism of action. I really tried very hard. No one in this world will tell you with such a clarity, with such an extent. You got my point? One question pakka. One MCQ pakka from this. And if I tell you this 8 times alag-alag na, you will forget ki, oh ma'am, kisme kya tha, which one was having cyclic AMP, which one was having protein inhibition. I'm getting confused. So till the end, when I will teach you 30 bacteria, you will become mad ki which have which one. Now I have summarized. Now it will be very easy for you. Now this is the first time I told you, na? So the remaining 7 times, I will show you this mnemonic. I will show you these 2 mnemonics 7 more times. This is the first time I am telling you. I will show you today only. आज की तारीख में I will tell you 7 more times when the other 7 bacteria will come. Currently, it is for Bacillus anthrax. I told you 7 more times I will tell you. है ना? So this is the mechanism of action. You can see. So this is the toxin, Bacillus anthrax का toxin. It is going and increasing cyclic AMP. By increasing cyclic AMP, it is destroying the cell. ठीक है? So you can read the same thing. These are the 4 toxins. Delhi Private Secondary School. है ना? Diphtheria, Pseudomonas, Shiga-like. They inhibit. You can see the bacteria is not going. The toxin is going in the human cell. The toxin is inhibiting elongation factor 2. No elongation factor 2, no ribosome, no protein synthesis, cell death. So these 4 act by this mechanism. And these 4, be very efficient boy. Be very efficient boy. These 4 increase cyclic AMP. By increasing cyclic AMP, they destroy the cell. Ultimately, ये भी cell को destroy कर रही हैं, ये भी कर रही हैं, but the mechanism is different. My point is that. Say yes. Okay. So I was teaching you Bacillus anthrax. There are 3 types of anthrax. They are in front of you. This is cutaneous anthrax. This is pulmonary anthrax, and this is intestinal anthrax. है ना? Cutaneous occurs by wearing animal. Maybe this lady have wear a scarf, है ना? Scarf, and the animal is infected, है ना? And pulmonary occurs by inhaling the spores, and intestinal occurs by eating animals. है ना? We know the ways. ठीक है? So please learn the symptom. In cutaneous, ischar, a black ischar is formed. Black color ischar is formed. It is known as ischar. Ischar का मतलब होता है, it's a Greek word. Ischar का मतलब है कोल. कोयला काला है. सुना है song? कोयला काला है. So it is black. Because it is black color, that's why it is known as ischar. ठीक है? So black ischar is always there. And it is also known as the black ischar. The black ischar also known as malignant pustule. Malignant pustule. Pustule, whatever is the spelling, है ना? It's misnomer. It's not malignant. इसको बोलते हैं malignant, but it's not malignant. So learn the other name of this ischar, है ना? It's malignant pustule. In pulmonary, you can see the patient have the pneumonia and hemothorax. Hemothorax, blood is filled and person die. It's very lethal. It's very lethal. Person die. और intestinal वाले में diarrhea वगैरह हो जाएगा, है ना? It's diarrhea. So there are 3 types of anthrax. How many of you got it? We are done with this bacteria. है ना? I'm teaching you gram-positive bacilli. I have to teach you 6. Out of the 6, A, B, C, D, L, M. मैंने B complete कर दिया. 5 और हैं. jaldi, fatafat, 5 more. Say yes. Bacillus anthrax. So what I taught you in Bacillus anthrax? Listen, I taught you number 1, you have to tell me the morpho. Comment on morpho. Say something about capsule, say something about spore, say something about motility. That was morpho. है ना? उसके बाद tell me the culture media. ठीक है? In the culture media, you have to tell the nutrient agar, you have to tell the gelatin swab, you have to tell the solid media containing penicillin, you have to tell with methylene glue and the selective media. ठीक है? कल्चर मीडिया हो गया है ना? तुम्हें लर्न नहीं करना है. द क्वेश्चन विल बी इन फ्रंट ऑफ यू. ऐसा नहीं है कि राइट ए शॉर्ट नोट ऑन कल्चर मीडिया. ऐसा नहीं आएगा क्वेश्चन. दे विल आस्क यू समथिंग स्पेसिफिक. ठीक है? उसके बाद व्हाट इज द मैकेनिज्म ऑफ एक्शन ऑफ टॉक्सिन. In one word you have to tell me the mechanism of action of the toxin. ठीक है? Mechanism of action of the toxin. And then you have to tell me the diseases, the 3 type of diseases. है ना? So tell me the diseases. So start seeing people. This is the chapter we have done, completed. On one page we will revise Bacillus anthrax with me. है ना? If you people will be interactive, I will be with my this energy. If you keep quiet, I will also sit like this and ठीक है? Syllabus ही complete करना है ना? I will flip the pages. You want me to be energetic, you have to be energetic. Okay जी? चलो morphology फटाफट. Morphology में capsule present or absent? Say ma'am capsule present. Spore present or absent? Say ma'am spore present. But motility is absent. So tell me one unique property of capsule. What is the unique property in that? The unique property is that it is made up of polypeptide, not polysaccharide. It's polyglutamate, to be specific. Spores are there, but say ma'am they are non-bulging and central. They are not bulging, they are non-bulging and central. So these are the unique properties. On nutrient agar, it's medusa head. ठीक है? On gelatin swab, it's inverted fur. On solid media containing penicillin, it's string of pearls. On methylene glue, it's Mac-Faridian reaction. And selective media is PLET. Please learn thoroughly. Mechanism of action of the toxin in one word, you say they increase cyclic AMP. The toxin increase cyclic AMP by stimulating an enzyme known as adenyl cyclase. This is the mechanism of action. And it causes a disease. The name of the disease is anthrax. Disease तो एक ही है anthrax, but anthrax is of 3 types. It can be cutaneous anthrax, it can be pulmonary anthrax, it can be intestinal. Cutaneous में ischar is formed, malignant pustule is formed, pulmonary में pneumonia occur, and intestinal में diarrhea occur. Okay, very good. Everyone is good. Excellent. You people are too good. I'm proud of you. So this is the complete Bacillus anthrax. You will be able to solve all the question. Read the question, tell me the answer. Chalo, read the question. Inverted fur appearance on gelatin swab is a characteristic of which bacteria? What is the correct answer? Yes, inverted fur appearance on gelatin swab. Inverted fur appearance. So obviously, the correct answer is Bacillus anthrax. So I can change the question 5 times. I'm not changing it. You know. So one time I will do medusa head. Answer is same. I will do string of pearls. Answer is same. And a Mac-Faridian reaction. Answer is same. So these all are your PYQs. Please learn them. All 5 का PYQ. Next question in front of you. There is a person working in an abattoir. What is abattoir? Butcher. Abattoir मतलब where the animals are cut. It is a place where we cut the animals. है ना? So there is a person वो abattoir में काम कर रहा है. Abattoir is the biggest clue. They are talking about some zoonotic disease. है ना? Abattoir में किसी को कोई disease हुई means they are talking about zoonotic disease. है ना? They are talking about zoonotic disease. Presented with a pustule on the hand, the malignant pustule on the hand, and which later on turns into the black ulcer. So which stain will you use? First make the diagnosis. Obviously the diagnosis is Bacillus anthrax. I got 2 clues. Number 1, the person is an abattoir, and number 2, the person is having a pustule. Based on which my diagnosis is Bacillus anthrax. So Bacillus anthrax को final करने के लिए मैं कौन सा stain करूं? Methylene glue, carbol fuscin, acid fast और Calicor white. So obviously polychrome methylene glue जिस पे मुझे मिल जाएगा Mac-Faridian reaction. So it's an indirect question. It's not a direct question. Yes, it's an indirect question. And based on which we give the answer. One more side hero. I told you I'm teaching you Bacillus. So main hero is done. Bacillus anthrax is done. Now one side hero, Bacillus cereus. Anna, Bacillus anthrax causes anthrax. The 3 type of anthrax. Anna, cereus causes food poisoning. Cereus causes food poisoning. Learn the 2 differences. Bacillus cereus, it causes food poisoning. वो तो ठीक है. Anna, Bacillus anthrax was non-motile. This is motile. Bacillus anthrax was capsulated. This is non-capsulated. See. Please see. Bacillus anthrax, Bacillus cereus. है Tell me the regarding capsule. Tell me regarding motility. Who will tell? Capsule का क्या चक्कर है? Capsule का क्या चक्कर है? This one is capsulated, a unique capsule, polypeptide wala. This one is non-capsulated. But this one don't have the motility. This one is motile. Okay. क्यूट बेबी स्लीप में बी इज बेसिलस एक्सेप्ट एंथ्रैक्स. एंथ्रैक्स छोड़ के बाकी सब मोटाइल ही हैं. बाय पेरिट्रैकस फ्लैजिला. याद हुआ क्या? सो यू कैन गेट क्वेश्चंस ऑन दैट, है ना. दिस कॉजेज एंथ्रैक्स which is of 3 type, है ना. Cutaneous, pulmonary, intestinal. This causes food poisoning, which is of 2 type. I will tell you the 2 type of the food poisoning. It causes 2 type of food poisoning. Got it? So the disease is the food poisoning. Listen, what are the 2 type of food poisoning? In one food poisoning, only vomiting occur, no diarrhea. It is known as emetic type. Only vomiting, no diarrhea. In other food poisoning, only diarrhea, not vomiting. Got it? So there are 2 type of food poisoning, Bacillus cereus, है ना. Here we have only vomiting, no diarrhea. Here we have only diarrhea, no vomiting. Got it? The 2 type. You will see, ma'am, what is the difference between them? Okay. Let me draw the 2 humans to explain you the 2 type of food poisoning. Let me, just a second. The same I would like to draw again to explain you the 2 type of food poisoning caused by Bacillus cereus back to back. I know this person, the first person is eating what? The first person eating fried rice, Chinese fried rice. You like Chinese fried rice? I don't like any Chinese food. Anyways, fried Chinese rice. Okay. The second person is eating the vegetables. The second person is eating the vegetables or meat contaminated, है ना. So here the person is eating the toxin. Both of them have Bacillus cereus toxin, है ना. So here the toxin will go inside. The toxin will go inside from the blood. I mean from the intestine. The toxin will go in the blood. The toxin will go in the blood. From the blood, it will go to the brain, and in the brain, it will stimulate the vomiting center. So this person have only vomiting, no diarrhea. So fried rice leads to vomiting, only vomiting, no diarrhea. You have to learn. Only vomiting, no diarrhea. Okay? And it is within 2 to 6 hours after eating, within 6 hours. Anna, why it is very fast? Within 6 hours, the vomiting start. Why? Because the person is eating preformed toxin. The toxin is preformed in the Chinese rice. It is preformed only, preformed toxin. So person is not eating the bacteria, the person is eating the toxin. And toxin is preformed immediately after going, it is going to the brain and causing vomiting. It is known as emetic type. Say yes. The second person is eating the vegetable, meat. So here the bacteria will go inside. Toxin will not go, bacteria. Bacteria after going in the intestine secrete the toxin. And this toxin increases the peristalsis and it will cause diarrhea. Here only diarrhea occur, no vomiting. And it requires 6 to 16 hours. 6 to 16 because it will take time. By bacteria first it is going and then secreting the toxin. Say yes. So what are the 2 type of food poisoning? Can you tell me? Bacillus cereus की 2 type की food poisoning बताओ. What is the food type of food poisoning? 2 types. Say ma'am, emetic type and diarrheal type. ठीक है? In emetic type only vomiting, no diarrhea. In diarrheal type only diarrhea, no vomiting. Tell me the food. This occurs by Chinese rice, Chinese fried rice, and this occurs due to vegetables or meat. Okay, है ना. Incubation period बताओ. This is within 2 to 6 hours. This one 2 to 6 hours. This is 6 to 16 hours. Why it is very short? Why it is very long? Now say the reason. It is very short because here the toxin is preformed. And here it is long because toxin is formed. The toxin is formed in intestine. Toxin is formed in intestine. So you get 4 differences here. Learn first, second, third, fourth. The 2 type of the food poisoning. You get many questions on that. Say yes, say yes. So the same table is given in front of you. You can read. And same, the 2 type of the food poisoning. What is the selective media? Now, if it is vomiting, take vomitus as the sample. And if it is a diarrhea, take stool as a sample. है ना. यहाँ पे vomitus ले लो और यहाँ पे stool ले लो. So sample can be anything depending which type of food poisoning it is. Selective media. The selective media is MYPA. What is the full form of MYPA? MYPA. M for mannitol, Y for egg yolk, P is phenol red, and A is agar. है ना. MYPA. So this is the thing. Say yes, everyone. We are done with Bacillus anthrax, Bacillus cereus. We are done. है ना. So I told you this one is capsulated, this one is non-capsulated. This one is non-motile, this one is motile. This causes a disease known as anthrax, which is of 3 type, pulmonary, intestinal, cutaneous. This causes a disease, food poisoning, which is of 2 type, emetic and diarrhea. Okay, selective media of both of them. Here the selective media is PLET and here the selective media is MYPA. See the disease, see the selective media, see the morphology. This is how you compile the things. Okay, we are done with Bacillus. Okay. Okay. Staph aureus is also emetic. Harsh, I told you it is both. Staph aureus causes both. It causes diarrhea also, vomiting also. ठीक है? चलिए. So we go to the question. I guess question is very easy. Chinese restaurant syndrome after eating fried rice. Fried rice खाने के बाद कौन सा bacteria? Which bacteria is it? Chinese restaurant syndrome. So Chinese fried rice. Chinese fried rice. Obviously it's Bacillus cereus. Question is not very good. Question is not very good. Question was good if you have two options, Bacillus cereus. Bacillus cereus, one is emetic type and one is diarrheal type. Question was better. You can get confused. Abhi to bahut easy hai. So in that case also it is emetic type. Fried rice is emetic type. Okay. So can we go ahead? So we are done with one of the bacillus. Now I'm coming on the next, diphtheria. Can I start diphtheria? So let's start diphtheria. The next topic is diphtheria. So let me start with diphtheria, है ना? So A, B, C, D, L, M का B हो गया, अब D पे आ जाते हैं. 4 more after this, है ना? So I am expecting कि in the next 1 hour you can complete this. So after lunch we are left only with one category, gram negative bacilli. Miscellaneous are very small, we can complete. हाँ ठीक है, अगर रह भी गए आज तो कोई बात नहीं. But after lunch we have the big category to complete gram negative bacilli. ठीक है? So this is the thing. ठीक है? So before lunch let's complete this. Let's charge up. So let's come on the next bacteria, diphtheria. Corynebacterium diphtheria. Corynebacterium, Corynebacterium diphtheria. So the next bacteria is Corynebacterium diphtheria. Next bacteria. ठीक है? This diagram I have already explained you yesterday. Have I explained you this diagram yesterday? Say yes. Have I explained you? You can see it is which stain? See, ma'am, it's Albert stain. I told you yesterday Albert stain. Those who have attended my yesterday's lecture, they know what is Albert stain. Albert stain only 5 bacteria. Remember, जिसमें metachromatic granules होते हैं. कौन से होते हैं 5 bacteria? MHC गार्डन आगरा. याद आया? MHC गार्डन आगरा का जो C है वो है Corynebacterium diphtheria. So it is one of the bacteria stained by Albert stain. So this is Albert stain. So first identify which stain? Say ma'am, Albert stain. In the Albert stain, what is the color of the bacteria? Say green color. The bacteria is green color. Anna. And what is the color of the granules? The color of the granules is blue. It's dark blue color, the color of the granules. Okay. The bacteria is typically angulated in appearance. Can you see? The bacteria is angulated. It's either V or L. It's looking like a Chinese language. It's looking like a Chinese language. That's why known as Chinese letter appearance or cuneiform appearance. Have I told you this yesterday? So you get many questions on this. Diphtheria pe ye puri kahani yaad rakhna. Please learn the story on the diphtheria. You get questions on that. Coming on the culture media of diphtheria. So let me first summarize culture media of Corynebacterium diphtheria. है ना? I'm telling you the culture media of Corynebacterium diphtheria. ठीक है? Let's come on the culture media of Corynebacterium diphtheria. So there are 4 culture media. Number 1, Loeffler's slope. Loeffler's slope. ठीक है? There are 3 culture media. Loeffler's slope, this is not selective. The other 2 are selective. The other 2 which are selective is potassium telluride agar and the next is Tinsdale agar. Tinsdale agar. These 2 are selective. These 2 are selective. Loeffler's slope is not selective. You tell me what is the type of Loeffler's slope? Which media? I told you yesterday 6 media, na? Enriched, enrichment, transport, differential, indicator, selective. So Loeffler's slope kahan aata hai? Where does the Loeffler's slope come? Can anyone tell me? कल मैंने types of media पढ़ाए थे आपको. Yesterday I taught you the types of media. Very good, Prabingal. I told you it is a type of enriched media. It is a type of enriched, not enrichment. It's a type of enriched media. Enriched media is rich. It contains something additional. So either blood or egg or serum. So this contains serum. That's why it is an example of enriched media. It's not selective, it's enriched. And so one by one we will see all. So the first is the Loeffler's slope. On the Loeffler's slope, cream colonies are formed. Can you see the cream color colonies? But see, it's non-selective. If you wish, यहाँ पे लिख लो enriched. यहाँ पे लिख लो it's a enriched media. Bracket में लिख लो not selective. ये selective नहीं है. It's enriched. है ना? Now this is potassium telluride agar. This is potassium. This is selective. ये potassium telluride agar है. On potassium telluride agar, black color colonies are formed. Can you see? ये सारी black color की colonies बनी हुई है. So all the black colonies are formed. On potassium telluride agar, black colonies are formed. ठीक है? And I'm coming on that. ये जो 3 type है. On the last one, Tinsdale agar, it is also selective. Tinsdale agar पे भी black या brown colonies are formed. Either black or brown colonies. The black or brown colonies are there on Tinsdale agar. So Tinsdale agar is more selective as compared to potassium telluride agar. Tinsdale is more selective. It contains additional cysteine. इसमें additional हमने cysteine डाला तो ये और ज़्यादा selective हो गया. So पहले मुझे तीन culture media बताओ. Tell me the 3 culture media for Corynebacterium diphtheria. Who will tell? Tell me one non-selective and 2 selective. First enumerate it. One non-selective and 2 selective culture media. The non-selective one is the Loeffler slope. And the 2 selective, कौन से हैं 2 selective? 2 selective कौन से हैं? Potassium telluride agar and Tinsdale agar. Potassium telluride agar and Tinsdale agar. ठीक है, these 2 are selective. Tell me the color of the colony. Here cream colony, here black colony, and here brown colony. ठीक है, Tinsdale is more selective than potassium telluride. हालांकि दोनों selective हैं, but Tinsdale is more. Why? Because Tinsdale contain additional cysteine. Cysteine से additional सारे बैक्टीरिया मर जाएंगे. Only one will survive, that is Corynebacterium diphtheria. ठीक है. Now, let me tell you, Corynebacterium diphtheria is of 3 types. Corynebacterium diphtheria is of 3 types. The one which causes severe disease, the one which causes moderate, the one which causes mild disease. Corynebacterium diphtheria is of 3 types. है ना? The one which causes severe disease is known as gravis type. Gravis मतलब grave, बहुत severe disease. The one which causes moderate disease is intermediate type. And the one which causes mild disease is mitis यानी mild, है ना? तो मुझे पता करना है कि कौन से type का है. Just suppose I am having a specimen. I know it's Corynebacterium diphtheria. It is having Chinese letter arrangement. It is Corynebacterium diphtheria. But I want to know, is it severe, moderate or mild? ये तीनों में से कौन सा है? तो कैसे पता चलेगा? Is there is a way हम पता कर सकते हैं? We can make it out on PTA, potassium telluride agar. All 3 form black colony. Black तो तीनों बनाएंगे, लेकिन morphology is different. See, है ना? Can you see? This is a black colony, but it is looking like the daisy head. What is daisy? Daisy is a flower. Have you heard about the daisy? Google karo daisy. Like the rose flower, daisy. Daisy is a flower. So it's looking like a flower, sunflower के जैसा, daisy head. है ना? So it is gravis. Gravis form the daisy head. Okay, can you see? This is also black, but it is looking like the frog का egg. Intermedia is frog egg. है ना? And can you see this one? This one. It is looking like a poached egg. It is mitis. So you have to learn. Let me summarize. Finally मैं summarize कर रही हूँ culture media. है ना? Corynebacterium diphtheria का culture media. Corynebacterium diphtheria के 3 culture media हैं. There are 3 culture media. The first one is the Loeffler serum slope. The second is the potassium telluride agar, and third is the Tinsdale agar. Loeffler पे it form cream colony, PTA पे it form black colony, and Tinsdale पे it form brown colony. Among the 3, this one is enriched, and these 2 are selective. You can write if you wish. है ना? मैंने लिख के तो दिया है वैसे सब. Now PTA is most important. PTA के basis पे we can divide them into three types. है ना? Gravis, intermedia, mitis. ठीक है? Gravis is look like daisy head. है ना? Intermedia look like frog का egg. And mitis look like poached egg. I don't have any mnemonic, लेकिन उल्टा पुल्टा मत करना. Please. Gravis, intermedia and mitis. ये PTA पे हम 3 type. तीनों black हैं, लेकिन morphology is different. How many of you got it? I don't know, but I tried my best. So this is the culture media for the Corynebacterium diphtheria. समझ में आई क्या बात? Mechanism of action of toxin तुम बताओगे. You tell me. So this is Corynebacterium diphtheria. The Corynebacterium diphtheria secrete a toxin. It will secrete a toxin. The name of the toxin is diphtheria toxin. You tell me diphtheria toxin का mechanism क्या है? Human cell के अंदर वो जाके क्या करेगा? I told you the 2 mechanism. Let me give you 2 options. Will it increase cyclic AMP? Say yes or no. Or will it inhibit protein synthesis? Say yes or no. दोनों में से क्या करेगा? सेल के अंदर जाके. डिप्थीरिया टॉक्सिन कहाँ आ रहा था? डिप्थीरिया टॉक्सिन कहाँ पे आ रहा था? कौन से निमोनिक में आ रहा था? I told you 2 निमोनिक ना, DPSS or be very efficient boy. Yes, very good. It is inhibiting the protein synthesis. So no need to say. है ना? You can see this is bacteria. Bacteria is secreting the toxin. Toxin is going inside the cell, not bacteria. Toxin after going inside the cell inhibiting elongation factor 2. No elongation factor 2, no ribosome, no protein synthesis, cell will die. है ना? So I told you 2 mnemonics ना, DPSS का D है ये डिप्थीरिया. मैंने बोला था ना 7 बार और आएगा, अभी 6 बार और आएगा. This mnemonic. So I told you 2 mnemonic. DPSS, be very efficient boy. है ना? These 4 inhibit protein synthesis and kill the human cell. And these 4 increase cyclic AMP and kill the human cell. So I already told you Bacillus anthrax. I already told you diphtheria. The 6th time more it will come. समझ में आ रही है क्या बात? है ना? I'm trying to super simplify it. ठीक है? Now let me tell you the disease diphtheria. Can you see I'm drawing a human here? ओके ऐसे नहीं जाते. Let me draw a human body. ठीक है? And this is the blood vessel. ठीक है? Now, how does diphtheria enter in human body? How does diphtheria enter in human body? Diphtheria enter in human body by inhalational route. So it is entering via inhalational route, the droplets. After that, it is going to the tonsil. Diphtheria go to the tonsil. In the tonsil, it will cause necrosis of the tonsil membrane. It will cause necrosis of the tonsil membrane and it will produce a pseudomembrane there. By causing necrosis of the tonsil की membrane, diphtheria causes so. Corynebacterium diphtheria bacteria causes so. Corynebacterium diphtheria नाम का जो बैक्टीरिया है, it produces pseudotonsil. I will show you the image also. This is known as pseudomembrane. Pseudomembrane over the tonsil. Tonsil के ऊपर क्या बन जाएगी? Pseudomembrane over the tonsil. ग्रे कलर की, ग्रे कलर pseudomembrane tonsil के ऊपर बन जाएगी. After that, please mind my words. Please mind my words. The bacteria is producing the membrane. Bacteria will not go in the blood. Bacteria secrete toxin. Toxin will go in the blood. Okay, toxin will go in the blood. Bacteria will not go in the blood. The toxin will go in the blood. ठीक है, toxin have affinity for 3 organs. Number 1, heart. Number 2, adrenal gland. है ना? Adrenal gland. And number 3, nerves. The nerves coming from the brain. So the toxin will damage the heart, cause myocarditis. Damage the adrenal, causes hemorrhage. Damage the nerves, causes neuritis. This is known as diphtheria. The disease is known as diphtheria. है ना? What is the mechanism of action in the 3? So in the heart cell, in the adrenal cell, and in the nerves cell, it is inhibiting the protein. It is entering and inhibiting the protein by inhibiting ribosome. So heart is non-functional, adrenal is non-functional, and nerves are non-functional. The disease is known as diphtheria. तुम मुझे बताओ diphtheria कौन करा रहा है? Bacteria के toxin. You tell me. Who is causing the diphtheria? Bacteria is going inside and causing it, or bacteria secreting toxin? Toxin is causing it. So yes, it is caused by toxin. So can I say diphtheria is toxemia? Diphtheria is toxemia. Diphtheria is not bacteremia. Diphtheria में bacteremia नहीं होता है. Write down. Diphtheria is not bacteremia. Diphtheria is toxemia. Diphtheria में toxin जाता है blood में. Bacteria नहीं जाता है. Highlight करो. चिल्ला चिल्ला के बोलो. Diphtheria is toxemia. Diphtheria is not bacteremia because toxin is going and causing the disease. So you tell me treatment. ये person तुम्हारे पास आया, इसको treatment क्या दोगे? Let me give you 2 options. Antitoxin is treatment of choice or antibiotic is treatment of choice. What you will offer? Antitoxin ki antibiotic? I'm asking. What do you want to give? Antibiotic ki antitoxin? So of course, the treatment of choice is antitoxin. Antitoxin will neutralize the toxin. I know, but you will give antibiotic also. Because if you give the antibiotic, the bacteria will be killed in the tonsil. And no bacteria, no more toxin will come. Antibiotic is for prevention. भाई फिर से और नया toxin ना आए. Because if you kill the bacteria, no more new toxin will come. And whatever toxin already present in the blood, you have to neutralize with antitoxin. So you give both. You give antitoxin, antibiotic, both. But treatment of choice is antitoxin. Treatment of choice is antitoxin, not antibiotic. Okay? Antibiotic, we give erythromycin here. Yes. So can we proceed? Got it? So this is diphtheria. The same story is given to you. You can see this is, I'm talking about the white color membrane. Can you see the gray membrane? White or gray membrane in the tonsil. Appreciate the membrane. Because the pseudomembrane is formed, the neck will swell. The neck will swell. It is known as bull's neck. It is known as bull's neck. I know. So membrane is formed. ठीक है? Got it? Say yes. Got it? कोई confusion है? Last thing. Listen, there are 2 types of diphtheria. Then I will summarize diphtheria and we will solve the question. Diphtheria थोड़ा सा बड़ा bacteria है. Last thing. There are 2 types of diphtheria. This is a diphtheria. This is the nucleus of the diphtheria. This is the DNA of the diphtheria. Okay, this is the DNA of the diphtheria. ठीक है? The first diphtheria have tox gene. Tox नाम का एक gene है that produce toxin. The second bacteria don't have tox gene, so no toxin. And toxin, we have seen toxin ही disease कराता है. बिना toxin के disease नहीं होता है. Disease occurred only by toxin. Without toxin, we cannot cause the disease. So you can say this is toxigenic. Toxigenic strain. So it is pathogenic. It causes the diphtheria. It is pathogenic. This one is non-toxigenic. It is not. It don't have toxin. It cannot cause the disease. It is non-pathogenic, avirulent. It cannot cause the disease. Quick confusion. Quick confusion. There are 2 types of diphtheria. The one which have the toxin is pathogenic. The one which do not have the toxin is non-pathogenic. Say yes. हाँ तो बोलो. Can I convert non-pathogenic to pathogenic? Non-toxigenic to toxigenic? Is there a way? Is there a way we can convert? Say yes or say no. Is there a way? You will say, ma'am, we can convert. If this bacteria get the tox gene, कहीं से भी इसको tox gene दिलवा दो, तो it can do so. तो tox gene कौन देगा इसको? कौन देगा tox gene? So there is a phage known as beta phage. क्या नाम है उसका? बीटा फेज द वायरस. Yesterday I taught you the genetics now, the 4 ways. याद है क्या? ट्रांसफॉर्मेशन, ट्रांसडक्शन, conjugation and lysogenic conversion. याद आया क्या? So this beta phage have own tox gene. So it is not pick and drop service. It is not Swiggy, Zomato. Beta phage is giving own tox gene to this bacteria. So this non-toxigenic strain will convert into toxigenic strain, and this is known as lysogenic conversion. So via lysogenic conversion, we can convert non-toxigenic to toxigenic. इस पे भर भर के क्वेश्चन आता है. Via this lysogenic conversion. It's not written here. You can write if you wish. समझ में आई के बाद? So it's lysogenic conversion. समझ में आया? Huh? So what is the answer? How you convert a non-toxigenic strain to toxigenic strain, non-pathogenic strain to pathogenic strain in diphtheria? The answer is lysogenic conversion. Via lysogenic conversion, we can do so with the help of a beta phage. Beta phage will give tox gene, and with the help of tox gene, non-toxigenic will convert into toxigenic. Answer is lysogenic conversion. इस पे बहुत क्वेश्चन आता है. Highlight it, mark it. Got it? One more thing, last thing. I'm having a sample. I'm having 2 samples of diphtheria. It is of diphtheria only. I do not want to know कि diphtheria है कि नहीं. मुझे पता है it is Chinese letter, it's diphtheria. This is also Corynebacterium diphtheria. This is also Corynebacterium diphtheria. But one of them is toxigenic, one is non-toxigenic. One of them is toxigenic having tox gene. One of them is non-toxigenic not having tox gene. I do not know which one is which. मुझे पता करना है whether it is toxigenic or non-toxigenic. समझ में आया? हाँ? So I want to know the toxigenicity. Is there is a test? कोई test है? मैं apply करूँ? What is the test? Which can differentiate the toxigenic strain from non-toxigenic strain. बहुत easy है. So there is a test known as Alex gel. कल मैंने बताया था ये नाम आपको, Alex gel precipitation test, है ना? Alex gel. इसमें क्या करेंगे? Take up potassium telluride agar. You can see this is potassium telluride agar या blood agar. Take a rectangular filter paper. Rectangular filter paper cut कर लो, है ना? And dip it in the antitoxin. Dip it in the antitoxin. Antitoxin is available commercially. ये मार्केट में मिलता है. Antitoxin of diphtheria. Dip it and keep it here. Keep it here at the center. ठीक है? Now take your 2 strains one by one. Take this also, make a perpendicular streak like this, and take this also, make a perpendicular streak like this. है ना? One of them is toxigenic, one of them is not. मुझे नहीं पता कौन सा क्या है. So I'm taking the perpendicular streak. अब मुझे बताओ, where is antitoxin? Antitoxin will diffuse like this everywhere. Rectangular चारों तरफ से, all 4 side, it will diffuse like this. It will diffuse like this. Antitoxin हर जगह जाएगा. And one of the strain is producing toxin. मुझे नहीं पता ये A है ये B है. Imagine A is secreting and B is not secreting. तो इसमें से toxin निकलेगा. This is toxigenic ना, तो लेकिन इसमें से नहीं निकल रहा toxin. तो wherever toxin and antitoxin will meet, the bands will form. The bands of precipitation will form. So the strain which is giving bands of precipitation, it is positive. The strain which is not giving bands of precipitation is negative. Say yes. This is Alex gel precipitation test. Can you see the diagram? इस diagram पे 4 question. First identify the test. Test identify कर पा रहे हो? देखो diagram, इसमें filter paper है center में, containing antitoxin. क्या नाम है test का? Say ma'am, it's Alex gel. Alex gel. Second question, किस बैक्टीरिया के लिए करते हैं? Your answer is ma'am, Corynebacterium diphtheria के लिए करते हैं. Third question, किस चीज के लिए करते हैं? Corynebacterium diphtheria में utility क्या है? To know whether it is toxigenic or it is non-toxigenic. To know that we are applying this test. है ना? And जहां पे भी आपको bands मिले, देखो यहाँ पे इस वाले में bands हैं. I'm getting the bands here. So this one is toxigenic, and I'm not getting any bands here. In this one, I'm not getting any bands. है ना? So this one is non-toxigenic. So look at the bands. How many of you got it? I don't know. The one who got it, give me a thumbs up. Everyone. We are done with Corynebacterium diphtheria, by the way. ठीक है? So let me summarize Corynebacterium diphtheria. Corynebacterium diphtheria मैंने पढ़ा दिया, उसको summarize कर देती हूँ, है ना? First tell me morphology. सबसे पहले morphology देखेंगे हम. Please everyone. हाँ? सबसे पहले I will teach you the morphology. ठीक है? उसके बाद culture media. You tell me the culture media. Morphology में you have to tell me the stain. कौन सा stain हम करते हैं? ठीक है? उसमें appearance क्या होता है? Color क्या होता है बैक्टीरिया का? और appearance क्या होता है? ठीक है? कल्चर मीडिया में आपको बताने 3 कल्चर मीडिया. लॉफलर पे क्या मिलेगा? कौन सा कलर? पीटीए पे क्या मिलेगा? कौन सा कलर? और टिंसडेल पे क्या मिलेगा? कौन सा कलर? और इनमें से सिलेक्टिव कौन है, नॉन सिलेक्टिव कौन है? आपको ये बताना है. ठीक है? उसके बाद आपको बताना है मैकेनिज्म ऑफ एक्शन ऑफ टॉक्सिन. टॉक्सिन का मैकेनिज्म ऑफ एक्शन क्या है वो आपको बताना है. उसके बाद डिजीज बताना है. डिप्थीरिया डिजीज में क्या क्या ऑर्गन्स इन्वॉल्व होते हैं, सिम्टम्स क्या क्या होता है वो बताना है. और उसके बाद आपको बताना है टेस्ट ऑफ टॉक्सिजेनिसिटी. टॉक्सिजेनिसिटी का कौन सा टेस्ट है और हम कैसे अप्लाई करते हैं. सो दिस इज द समरी. आई हैव टॉट यू एवरीथिंग. यस, वेरी गुड. यू स्टार्टेड आंसरिंग. वेरी गुड, very good. So stain is Albert stain. The color of the bacteria is green color here, and the appearance is Chinese letter. Chinese या cuneiform. Very good. So you get many questions, image-based question from here. Culture media Loeffler पे cream colony बनती है, PTA पे black बनती है, और Tinsdale पे brown बनती है. So these two are selective. Loeffler is enriched, not selective. इनमें भी Tinsdale is more selective. है ना? Based on PTA, we divide them into three categories. ग्रेविस, इंटरमीजिया, माइटस. है ना? ग्रेविस में आपको मिलेगा डेसी हेड, यहां मिलेगा फ्रॉग एग या पोच्ड एग. ये भी लर्न करना है. मैकेनिज्म ऑफ एक्शन ऑफ टॉक्सिन अगर शॉर्ट में बोलना है, द मैकेनिज्म ऑफ एक्शन ऑफ टॉक्सिन, इट इनहिबिट प्रोटीन सिंथेसिस. वेरी गुड. इट इनहिबिट प्रोटीन सिंथेसिस बाय इनहिबिटिंग इलॉन्गेशन फैक्टर 2. डिजीज में पहले तो सूडो मेम्ब्रेन बनेगी, बुल नेक बनेगा, और 3 ऑर्गन इन्वॉल्व होंगे, हार्ट, एड्रिनल और नर्व्स. ठीक है? एंड दिस इज अ टॉक्सीमिया. This is a toxemia, not bacteremia. है ना? टॉक्सिजेनिक नॉन टॉक्सिजेनिक पता करने के लिए we can apply a test Alex gel. है ना? You have understood it completely. Anyone having any confusion? You are open to ask. Can we proceed? Anyone having any confusion? Can we proceed? Read the question, tell me the answer. Step by step approach करेंगे. Read the question, tell me the answer. What is the mechanism of action of the toxin produced by the bacteria shown in the figure? So first identify the bacteria shown in the figure. You identify the bacteria shown in the figure. So जब भी डायग्राम आए तो 2 चीज देखनी है, कलर और शेप. हमेशा स्टार्ट करो, रूल बना लो, जब भी डायग्राम आए, सी द कलर, सी द शेप. पहले कलर बताओ. रेड की वॉलेट, you all can see, you are not color blind. Red की वॉलेट, you will say ma'am it's violet, so it's a gram positive, and it's bacilli की cocci, so you can see ma'am these are elongated, these all are elongated, it's a gram positive bacilli. So first come on the conclusion कि है क्या. It's a gram positive bacilli, so answer A, B, C, D, L, M में से कुछ भी हो सकता है. I don't know. 6 में से कुछ भी हो सकता है. अब इमेज को और क्लोजली देखो, तो I got the angulated appearance. मोस्ट ऑफ द जगह यहाँ पे थोड़ा थोड़ा एंगल दिख रहा है. इफ यू सी इट क्लोजली ना, आपको एंगल दिखेगा. तो ABCD LM में D है, डिप्थीरिया. तो आंसर इज डिप्थीरिया. है ना? डिप्थीरिया की टॉक्सिन का मैकेनिज्म ऑफ एक्शन क्या होता है? साइक्लिक AMP होता है कि एलोंगेशन फैक्टर 2 प्रोटीन वाला होता है? कहां पे आ रहा था डिप्थीरिया? आई टोल्ड यू 2 निमोनिक. DPSS कि we be very efficient boy. It is DPSS. DPSS का D है. So it inhibit elongation factor 2. It inhibit the protein synthesis. So correct answer is C. How many of you got it? It's a double triple level question. पहले तो diagnosis बनाओ based on the image. Then tell the mechanism of action of the toxin. Okay जी. So correct answer is C. Say yes. It is a ribosylation of elongation factor 2. ADP ribosylation of elongation factor 2. Okay. So correct answer is C. है ना? Next question. You will tell me. पोस्ट एग अपीयरेंस. ग्रेविस की, माइटस की, इंटरमीजिया की, मिनिमस. बोलो जी, बोलो. हु विल टेल? हु विल टेल? नन टिल नाउ. वेरी गुड. तो शिवांगी, ससिका, वेरी गुड. बी की सी. B की C. Some people are saying B, some are saying C. So I told you 3 things ना. Gravis, intermedia, mitis. कुछ निमोनिक बना लो. मेरे पास नहीं है. If you have some mnemonic मुझे share करो. I will share with everyone. Gravis form daisy head. Intermedia form frog egg. And mitis form poached egg. कुछ निमोनिक बना लो. कुछ निमोनिक बना लो. Make some mnemonic. है ना? Gravis, intermedia, mitis हम इसी सीक्वेंस में लिखेंगे. Grave, फिर intermedia, फिर mitis. तो यहाँ पे DFP. You can make some mnemonic. Daisy head, frog egg, poached egg. So currently मैं poached egg पूछ रही हूं, तो आंसर होगा mitis. Minimus कुछ नहीं होता, mitis. So answer is B. Answer is B. If I change the question from poached egg to frog egg, आंसर हो जाएगा intermedia. And if I change the question to daisy head, आंसर gravis हो जाएगा. How many of you got it? So we get questions on that. So we are done with bacillus, with diphtheria. How many of you are with me and energetic and awakened and live? How many of you? Can we proceed? हाँ? So 6 में से 2 हो गए, तीसरे पे आ रही हूँ. है ना? तीसरा बस ये बड़ा है, बाकी सब छोटे छोटे हैं. The rest are small. ये वाला थोड़ा सा बड़ा है, Clostridium. So we will complete Clostridium also. So can I start the next Clostridium? Can we start? चलो, next Clostridium. Clostridium के बारे में आपको क्या पता है? First you tell. Clostridium के बारे में आपको कितना पता है और क्या पता है? आप बताओ पहले. कहाँ याद आता है Clostridium? You tell me कैप्सूल है कि नहीं है? अच्छा चलो. स्पोर फॉर्मिंग बैक्टीरिया कितने होते हैं? You tell me क्या निमोनिक है स्पोर फॉर्मिंग का? स्पोर फॉर्मिंग बैक्टीरिया आर BSc केमिस्ट्री. BSc केमिस्ट्री में one of the C is Clostridium. So you will say मैम ये स्पोर फॉर्मिंग है. है ना, ये स्पोर फॉर्मिंग है। स्पोर फॉर्मिंग तो बेसिलस भी था, लेकिन बेसिलस के स्पोर थे नॉन बल्जिंग, यहाँ पे स्पोर आएंगे बल्जिंग, यहाँ पे सारे स्पोर्स आएंगे बल्जिंग, ठीक है। सो दिस इज द डिफरेंस, ठीक है। ओके ओके, गुड डे इज फ्राइडे, व्हाट यू आर सेइंग, मंडे नाइट। So what is the mnemonic you are saying, Bilal? D, F and P. So what is your mnemonic? DFP का क्या mnemonic बनाया? DF, अच्छा, you have written this also. Gravis, intermedia, mitus. So good day, GD is good day, ठीक है। Good day is the Friday. Good day is Friday, है ना। और ये क्या है, M और P का क्या करूं? हाँ, मंडे तो ठीक है, P से क्या करूं? पोस्ट डेक बनाना है ना, और बनाओ, ठीक है। GD इज गुड डे, IF इज फ्राइडे, गुड डे इज फ्राइडे, यहां तक तो समझ में आया। और MP का कुछ बना लो, गुड डे इज फ्राइडे। ओके, एटलीस्ट बिलाल इज ट्राइंग, गुड, आई लाइक इट। मेंटल प्रॉब्लम उस्मान, मेंटल प्रॉब्लम रियली। चलो ठीक है, गुड डे इज फ्राइडे और गुड डे फ्राइडे पे हो गई मेंटल प्रॉब्लम, ठीक है। चलो मेक इट समथिंग, कुछ तो याद करो, ठीक है, ठीक है। So good day is the Friday और मेंटल प्रॉब्लम, है ना। एटलीस्ट ट्राई, सो दैट एग्जाम में गड़बड़ नहीं होना चाहिए, है ना। अब तुम बताओगे तो हर बैच में अगले आई विल प्रोसीड। आपको जितने निमोनिक मैं बताती हूँ ना, all the mnemonics are shared by the students only, and that is proceeded generation by the educator, ठीक है। अच्छा, MP मध्य प्रदेश, आई एम फ्रॉम मध्य प्रदेश, इंदौर ओनली, ठीक है। चलो, गुड डे इज फ्राइडे मध्य प्रदेश, वॉट एवर यू से, ठीक है। चलो, सो लेट्स प्रोसीड, चले आगे बढ़े, है ना। सो वी आर स्टार्टिंग विद क्लॉस्ट्रीडियम। आई वाज टेलिंग यू क्लॉस्ट्रीडियम इज स्पोर फॉर्मिंग बैक्टीरिया। स्पोर होते हैं क्लॉस्ट्रीडियम में, क्योंकि स्पोर फॉर्मिंग तो 4 ही बैक्टीरिया होते हैं। BSc केमिस्ट्री, C फॉर क्लॉस्ट्रीडियम, it is a spore forming bacteria. Not only this, what are obligate anaerobe, है ना। obligate is very disciplined, strict, anaerobe, so they grow anaerobically. If you give them oxygen they will die. क्या mnemonic था? Can't breathe air. If you give them air they will die. If you give them air they will die, है ना। so C is clostridium here, so it's obligate anaerobe, है ना। so it's obligate. So obligate anaerobe को yesterday I taught you. What is the media for obligate anaerobe? You tell me. What is the media? Yes, very good, Kartika. What is the media? It's RCM, है ना, Robertson cooked me. So for Clostridium, we use RCM. So you learn Clostridium is a spore-forming bacteria. Clostridium is anaerobic. Learn करने की जरूरत नहीं है. It's visible in the mnemonic. I'm just correlating. It's visible. है ना, ठीक है। So I'm starting with Clostridium. चलो चलो, बहुत सारे mnemonic हो गए. Now come back. Everyone learned it. अब कोई गलत नहीं करेगा, ठीक है। चलो, let's start with Clostridium. These all Clostridium are there in your syllabus. You'll say, ma'am, what do you mean? इतने सारे Clostridium. I will teach you only 4. Don't worry. The ultra important only 4 we will study. लेकिन इतने सारे होते हैं. You should have a look. देखो, I told you RCM. What is RCM? Robertson Cooked media क्या होता है? So it's a media जिसमें एक test tube होता है. We have a test tube. The mouth of the test tube is covered with paraffin wax. Remembered? And it contains nutrient broth. And it contains मीट का पीस, इट कंटेन मीट का पीस, ऑक्स हार्ट मीट पीस, याद आया? इट इज ऑक्स हार्ट मीट पीस। यस्टरडे आई टॉट यू। मीट के अंदर क्या होता है? मीट के अंदर 2 चीजें होता है। लिसन लिसन, मीट कंटेन प्रोटीन, मीट कंटेन कार्ब्स। डू यू ईट मीट? एनीवन ईट मीट? सो यू मे बी नोइंग मीट हैव बोथ प्रोटीन एंड कार्ब्स। मीट हैव प्रोटीन, मीट हैव कार्ब्स, है ना। व्हाट डू यू लाइक टू ईट? यू लाइक प्रोटीन और यू लाइक कार्ब्स? सबकी प्रेफरेंस होती है ना, किसी को कार्ब ज्यादा पसंद है खाने में, मीठा पसंद है, डेजर्ट्स पसंद है। some like to eat protein. Clostridia also have a preference. If you are putting the Clostridia here, no, listen. If you are putting a Clostridia, there are 4 possibilities. Listen, first possibility, some of the Clostridia will come and degrade only protein, not carbs. These are known as proteolytic, है ना। They like only protein to eat. They are proteolytic, है ना। They are very health conscious. They are proteolytic. They only degrade protein, not the carbs. Some of them degrade only carbs, है ना। They like the sweets, desserts. They eat only carbs. They don't eat protein, है ना। And these are known as saccharolytic, है ना, saccharolytic. So some Clostridia are proteolytic but not saccharolytic. Some are saccharolytic but not proteolytic. Some are both, some of none, है ना। हमें भी तो 4 ही लोग होते हैं. There are 4 type of people. Some eat protein, some eat carbs, some eat both, some eat none. And also the same occurs with the Clostridia. Some eat protein. Now listen. How we come to know which Clostridia is liking what? Whenever from this meat piece, have a look on this meat piece. Can you see this is a meat piece? I'm highlighting, है ना? Whenever the protein is degraded, whenever you degrade only protein, not the carb, the meat piece become black in color. And whenever you degrade only carb but not protein, the meat piece become pink in color. So see the color of the meat piece and decide whether the Clostridia is proteolytic or saccharolytic. समझ में आई क्या बात? समझ में आई क्या बात? हाँ? Got my point? See the meat piece का color, everyone. See the meat piece का color here. See the meat piece का color here. So this one is red or pink, एक ही बात है. Red or pink. This one is saccharolytic. इसके अंदर saccharolytic Clostridium है. That is degrading the carbs. Okay, okay. And here you can see this is proteolytic. It's black in color. है ना? Here the meat pieces are black in color. Okay, so it's proteolytic. है ना? The meat pieces are black. So saccharolytic protein. So what are the four types? Say, ma'am, there are four types of Clostridium. Some are proteolytic but not saccharolytic. Some are saccharolytic but not proteolytic. Some are neither proteolytic nor saccharolytic, and some are both. है ना? So you have to learn this classification. You will say ki, ma'am, how we learn this classification? It's looking very difficult. Don't worry. Learn first, second, third category. Remaining all will fall in the last. You can do like that. है ना? So only proteolytic is chitinine. Only proteolytic. It will eat only protein. है ना? And none of them is cochlearium. इन दोनों पे बहुत question आता है. I mean to say तुम्हें classification तो learn करना पड़ेगा. है ना? You get questions. At least this and this category you learn. So at least you get one question. है ना? One organism है. So high chances you get question from that. Okay, got my point? Okay, can we proceed? So this is the classification of Clostridium. Out of them, I am going to teach you four. Which four? Purkinje, Tetani, Botulinum, difficile. Please now, please everyone concentrate. Hai na? Don't gossip. Now please concentrate. I am going to teach you four Clostridium one by one, 5 minutes each. Hai na? In the next 15-20 minutes, we will finish this chapter. So Clostridium. There are four in our syllabus that we have to continue in detail. Purkinje का दूसरा नाम लिख लो. Purkinje is also known as Welchii. Purkinje is also known as Welchii. Students even don't know कि ये एक ही है. Purkinje और Welchii is one and the same. They are the synonym. है ना? दूसरा is Tetani. तीसरा is Botulinum. चौथा is difficile. First you learn. मैं तुम्हें चार पढ़ाऊंगी. है ना? अभी पढ़ाऊंगी चारों. Listen, I call Purkinje as गद्दार. क्या बोलते हैं इंग्लिश में गद्दार को? For the students who don't know Hindi. मुझे नहीं पता क्या कहते हैं. ठीक है. Anyways, why I call him गद्दार? Listen. All the Clostridium are non-capsulated, but no, this is capsulated. It is going against the family. Who is a gaddar? Who goes against to the family? Who go against the community? Everyone is doing something else, but someone is doing against everyone. So that is the, yes. So yes, it's a traitor. Okay. So it is against everyone. Got my point? So everyone is non-capsulated, but it is capsulated. Everyone is motile, but it is non-motile. So in both the things, it is going opposite of its family. Everyone else in the family is non-capsulated, it is capsulated. Everyone in the family is motile, this one is non-motile. But for spore forming, there is no gaddari. They all have the spore. Without any exclusion, they all have the spore. समझ में आई क्या बात? I am teaching you four Clostridium. Purkinje, Tetani, Botulinum, difficile. Purkinje also known as Welchii. पहली बात तो है ना? Now comment for their capsule, motility and spores. I mean to say comment on their morphology. एक बार में morphology बोल के खत्म करो. एक बार में morphology बोल के खत्म करो. You will say, ma'am, they all are non-capsulated except Purkinje. You say they all are motile except Purkinje, है ना? And spores are present in all of them. Spores by default present in all of them. I'm sorry. I'm sorry. I'm really sorry. Give me a minute, Hannah. So in the spores now, there is no exception. But in capsule and motility, you have to see there is an exception. So one by one, we will see the four Clostridium. Okay? Or else let's take a break. After the break, we will continue with the Clostridium. Can we take the lunch break? 1 hour. Or you want a small break, 5 minutes. After that, I will settle and come back. Or you want lunch break? What do you want? Okay, then let's take lunch break and 3-5. I will be back. Okay, 1 hour. Okay, bye-bye.